You know, if there's a way to, I would prefer not to use my laptop for everyone's because I a week. Is there a way to put like a flash drive into this? I just can't get any of this.
I tried there. I don't actually see.
Oh wait, I do.
Yeah, anything you get up on screen will come back to me with the broadcast, so it's all good. Whatever way you do it, just to make.
I'm not gonna mess with that. I'm having everyone email it to me. Don't. No joke. That's my. No, I'm saying I can't figure the flash drive thing out, so I'm just gonna have everyone email it to me. No, I know that's fine, but I'm talking about the first session. They're not. They can just connect.
Yeah, see, this is what he's saying. Use this USB stick to transfer files to the PC. You can also use the USB part for the slided. Is this on? Is this? Oh, that's on. You can also use the USB part for the slide advance. I want to unplug this. Yeah, that's how you do. And then here. Have it on mute. You can also use the USB part. Yeah, see? Like if they so this is just, but it says you can. It doesn't. Well, first people, so we can do slided. Otherwise, they'll just have to be over here. Oh, did it do? So only basically. I think flickers. Otherwise they can just. Only from this. But they can. Unless they have that. But they'll have to get really close. So that's why I was gonna say, like, no, it's stuck in there. Hello. So let's see.
Hello?
Have a laser. It only. Oh, like I think. Sorry, that's what I'm trying to plug in there. Pull up this line. I got you. The other thing for people, I can just.
The one on the
Testing.
is my first name. And Kubendra instrument.
Yes, I do. I said it.
Okay,
backwards.
no,
Sorry,
no,
I was
okay,
just
okay. I
making
was like-
sure. I was just repeating it to
No,
myself.
no worries.
Yep.
It comes. Yeah.
Oh, it is. Which regardless though. There's a laser. This should work once you upload your talk. Do you have your talk on a drive or anything? Can you give that drive to me? Later. Yeah. Well, just. You're right. Hey Jay, is this the font you had?
That's just for the title.
Okay, I'm just making sure that this. Because if I save it, it doesn't. But it should be. So this presenter. How it shows.
Yeah,
Here,
but
I'll have
also
to.
been moving on the stage.
Okay. And then this. There is a mic here, but we can also like, we're gonna see really. Once this goes with this.
I would like to have 5-minute one for the end of after 10 minutes.
Sorry. I was just making sure that it was.
No, it's okay. Yeah, you said that once.
Yeah, so I didn't save it. I just.
Yeah, let it be. Open.
Yeah, that's what.
Yeah.
Here.
Are you keeping yours on the thumb drive? You can do something different for
Are they gonna?
Walk around.
Yes.
We can. No, I'm not gonna worry about this.
Okay.
It's too much.
That's it. No, just facing when I talk because I don't want to be.
I figured people would. I figured from this one. I'll do it.
I was just
Okay.
like,
We need to figure out how to, yeah, we need to mute this.
Testing, testing. Testing, testing. Hello, hello. Okay.
testing.
I think it might be.
Let's see. Testing. Sounds good.
Move
Yeah.
around a bit.
Welcome to my talk, guys. I think it's good, right? Yeah. Yeah. Yeah.
Good morning, everybody. Please quickly find a seat so we can get started on time for our very first session. So exciting. A quick housekeeping announcement. If you're presenting in an upcoming session, try to come up a few minutes early to make sure your slides look fine. If you didn't for the session, that's okay, because we're still going to get started and get rolling. If you didn't pick up your name tag yesterday, I will have those at the coffee break at 10, if I can remember. And Taylor's going to introduce our first session.
Okay, our first session is going to be hydrothermal vents, the jacuzzis of life. Thank Justin in the front row for that name. All right, our first speaker is Mrityunjay Kubandran Sumati, talking about the unveiling thermal adaptation in organisms, insights from an energy-centric perspective in astrobiology. Take it away.
Can you all hear me all right? Yes? Cool. So that's my title, and it was done with the help of ChatGPT. So if you guys want, you can ask nice titles. Do you guys like the weather at Cornell? Yeah? Good? But I bet if I ask you to put your hands in a boiling water, you would not, right? But then there are a bunch of microorganisms that it's not just they can tolerate these high temperatures, but rather they need this high temperature to grow. And those are the microorganisms I'm going to talk about, and I'm going to ask a bunch of questions and discuss more about them. So what is this temperature, and why is that such a big thing? So temperature affects everything in life, from physical to biological parameters. It decides how much of gases that's going to be soluble in any environment. And on the biological side, it's going to talk about how stable are the biomolecules going to be, at what rate are the enzyme reactions going to happen, and how fast can an organism move, and so on. So all the microorganisms that we find in hydrothermal vents or any other super hot systems, they are practically microorganisms that have overcome these kind of issues. And so overcoming these issues is rather simple. You know, it's going to get hot, and then you turn on your air conditioning, right? That's as simple as that. But there is one small problem. If air conditioning was never invented, and then I'm going to tell you, well, it's 85 degrees Fahrenheit today, but tomorrow it's going to be 105, would you be able to cope starting tomorrow? So probably not. But if you could, you can cope for a week, a month, but eventually you're going to die. So, but for microorganisms, this is going to be pretty fast. And so the question is, where did these adaptations come from? These adaptations that we see today, they definitely confer adaptation towards high temperature, but is that what drove their survival at high temperature? So I looked back at the ancestors, and if you guys are familiar with the last universal common ancestor, you know the debate about whether it was a hyperthermophile, whether it was a mesophile or a thermophile. But let's not look at Luca yet. Let's go back to the ancestors of bacteria and archaea. That's where we have consensus that they were definitely microorganisms that were already adapted to high temperature. So it's, in some sense saying that our ancestors invented air conditioning, and they passed down the information to us, and that's what we are using today. But then Luca, we have the debate. So in my study, to understand how any life would adapt to high temperature, I was stuck there, because it's like there is low temperature, high temperature. We don't know what Luca was. So where do I start? And then my PI said, why not do everything? So I said, okay, let's assume that Luca was a low temperature organism. So what happened was it went from low temperature to high temperature when it became the ancestor of bacteria and archaea, and then went on to adapt to all these other environments. Or if it was a high temperature microorganism that went on to be the ancestral high temperature microorganism and adapted to other environments. This makes me ask three questions. how did low temperature microorganisms adapt to high temperature? And how did high temperature microorganisms change themselves as they survive lower temperatures? And finally, why don't we find today's organism crossing this temperature barrier and going to high temperature? And this is important because most of the experimental evolutionary studies where they grow these microorganisms at higher and higher temperature, they all come to a standpoint. They cannot push this barrier further. So what is stopping this thing? And as I just said, experimental evolution is not going to take us far. So this is where I come into. I said, you know what, how about if we try modeling? Let's try to create models, particularly evolutionary models, which is going to allow us to test evolutionary strategies. Can this organism evolve a set of traits that would adapt at high temperatures? So some conditions we need for the model is that I'm talking about LUCA and all microorganisms and probably all life. So the model that I'm making has to be general, and it should be applicable for all microbes and all life. And it should also determine the thermal boundaries as to what would be the lowest limit be for that organism, or what would be the highest limit. And you have a bunch of other requirements the model should fulfill. This is where I present to you. So come with me on this journey, and I have 3 important slides, basically. So that's all I'm trying to convey here. This is the first slide, explanation of the model. So we know that all microorganisms need energy, and that's constant. And be it carbon-based life or even silicon-based life or any other life, life means that you move yourself out of equilibrium and stay in that situation. So the way life on Earth works is that it takes substrates, converts them into products called catabolism, produces free energy, converts that into ATP molecules, which can then be used for anabolism. Now, what happens in anabolism is that you could spend your energy that you get into 2 different ways. One is to maintain yourself, which is very important, because if you cannot fulfill that energy, you're going to die, basically. And the second thing is cell division. And the way we are modeling that is that since you need to be alive first, you will have to fulfill this maintenance energy. And whatever energy you have after that is what is going for growth. So you have, let's say, a very little energy after your maintenance needs, you're going to grow very slow. But if you have a lot of energy, you're going to grow quickly. And this is what we see across E. coli, which has 20 minutes lifetime, or for those microorganisms that live in deep sea hydrothermal vents, which have 5,000 years of one cycle. So what does this tell us? By using this concept, we can have a plot like this that explains biology, basically. The green line here is basically at what rate you're going to get your catabolic energy in. So the way the green curve is shaped is basically a simple enzyme kinetic curve. As you increase the temperature, you're going to reach an optimum condition where you're going to be the fastest at getting the energy out, and beyond that, your energy is going to decrease. And the orange curve is how much it costs to maintain yourself with regards to temperature. And studies have said that it should follow some sort of an exponential increase. Now, if you start with a very low energy, this is how it should increase. But if you're going to start with very high maintenance costs, you would have a plot something like this, which increases rather quickly. And the idea here is you could just do, it's 2 power x as a simplest model, and you could be if you start at 2, you're going to take, to reach 2 million, you're going to take much longer than if you start at 20. So with this idea, what we have is that this space within which the energy that you get is higher than the cost that you need to provide. So you can only survive in this space. And all the space outside here, your maintenance cost is way higher than what you get from the environment. So no survival there. And these points are the points where the limits. So as I said, this model can tell me the limits, survivability, and within this space, it becomes more of an optimization problem where the cell just has to optimize itself to its maximum difference so that it could grow the fastest. So this is what the overall outline of this model is. So and then I went on to see, okay, I have this model now, but how are these microorganisms that are already at high temperatures doing, working with these kind of models with their energy? And what I find is that most of the microorganisms that are at high temperature, particularly above 90 degrees Celsius, are only archaea. So there must be something special about them, right? And what is special and what has been found previously is that archaea tend to reduce their maintenance cost. As I said, if you start at a lower maintenance, you're going to start lower and increase the cost way slower. And that's what archaea are doing. So I was like, well, that makes sense because now I have some form of evidence for that my thinking is correct. So then I asked, okay, so if archaea reduce their maintenance cost, what contributes to this cost? And depending on the different situation, this could be different. But these are the key pathways that you need to have in a cell, which is taking in the substrate, catabolizing it, gene regulation, making ATP molecules, making the proteins, making the enzymes that needs to be there, doing all the other functions. So, and what is interesting is that all these things are functions of enzymes. Everything uses enzymes. Almost everything uses enzymes in Earth. So increasing the number of enzymes is like starting with a higher maintenance energy. So that's the first hypothesis I was thinking. So if a cell is going to have a super complex network of biochemistry, then it's going to start at a much higher maintenance energy, and it's going to go faster. So then I asked, so how do all of these relate? And this is another plot how I'm going to tell you how the evolutionary cycles could be, and we're going to shift between these two plots. So consider this moderately temperature microorganism with these survival space. So if it wants to go to a higher temperature, so what it has to do is decrease its maintenance energy, so thereby moving this red line slightly lower. Now you have this new blue space that this life can occupy. And that green spot over there is a much higher temperature than it originally was or could. So then its optimization problem, it moves these curves by making sure that these enzymes are more thermostable and now has this new high temperature space where the microorganism could grow. So that's how it goes as yellow and then turns as red. So if you do this, you could have a space like this. And this is all hypothesis. This is all what I thought it could be working like this. And usually what happens is that at lower temperature, your temperature stress is alleviated, which means that you can access much more options. You could go for different metabolisms, different sugars, highly complex sugars, and so much, and increase your complexity. So this is what I thought could be happening with this life. And this white space that you see would rather be a space that is inaccessible for any life. And the reason is because in this space, if you have a complexity that's going to be super high to adapt to such high temperatures. And so I asked two questions. Is it true that these microorganisms that live at higher temperatures shift their maintenance energy curves lower? And do complexity vary with temperature? And the first result is yes. And this is raw data, so it's not super clean. But what you see is that microorganisms, the more they go towards high temperature, they are kind of shifting their maintenance curve lower and lower, like the example I showed you. And for the second test, I analyzed complexity for around like 6,000 species, and I ended up with 3,000 samples. And what I find is that complexity sure is negatively correlated. And as you go higher up at temperature, you're going to be lower and lower complex. What was really interesting for me is that I found the same trend, the inaccessible space, happening in all these slides. This white space where we don't find any life on Earth, suggesting that there is much stronger correlation to what I was proposing and the data that we are observing. And these are strong correlations that I find, not causation. So we have to test them and we have to give them proofs. And this is where I am going to end, where you can have this kind of idea and these curves written up as a simple model where this is the green curve, this is the red curve, and this is the ultimate growth. And in an evolutionary model, what we do is we say, okay, there is a mutant that can change these conditions. And if such a mutant can come up, can that change the growth rate and succeed in that high temperature environment or not? So that's the evolutionary model that we are going to test. And what's the significance of this is that this model I'm developing in context of temperature. But if anyone is interested in pressure, you could rewrite all these parameters as a function of pressure and do the same plot for pressure and understand how any life could behave with regards to temperature. And remember, this is an evolutionary space. So, which basically means that it is only within the space the microorganisms can evolve and adapt. You cannot go outside of this space. So, and one more extra cool thing is that it just doesn't have to work for carbon. It can also work for silicon-based life. If you can find what is the biochemical reaction that silicon-based life would bring you energy, and what is the biochemical reaction that silicon-based life would use to build itself up, and there you have the parameters of this equation, and you could get a similar plot. So this could inform a huge number of things about astrobiology and also help us understand how microorganisms adapt and evolve in general. And with that, I would like to thank you, and I would like to take any questions. Yes. This one? So this is a graph which we call pairwise invisibility plot, which is what you use to test your evolutionary models. And the idea, well, in the simplest way, it's really complicated for me to explain now. But the idea here is that you can find different of these points. They could be either stable points and attractors or stable points and repellers. So which means that if I have a, say, the best way to explain is, okay, this one. If I have a mutant over here, and if this center point is a stable point and an attractor, it means that any mutant that could have a better strategy is going to go towards the steady state. So which means you can keep pushing yourself to find the boundary. But if it was something like a repeller, then what you could just have is that no matter what mutations you can have in genes or what changes you could do in a cell, you will never cross that boundary point. So that's talking about various all the different parameters that you could use, but it would ideally be like, for this plot, you would get one pairwise invisibility plot, which is just one of these squares. And then you will analyze that further. Yeah. Yeah. So again, it's the dynamics between the maintenance cost. So at high temperature, if you are more complex, it means that you need more maintenance cost. But the rate at which you get the energy from the environment, it cannot just be fast enough for you to satisfy your maintenance needs. And that's why you're going to be like, you're going to basically starve at that temperature, where what usually would happen, and when we go very close to thermodynamic limits, is that you will often find like a decoupling, where your energy is going to come in, but that's just not enough for you to sustain yourself. So you cannot, like, it's something like, if we take some fat loss regime where it's like we cut down the diet, so and the reason is that our fat is being burnt out. So if you keep doing that, you're going to starve and you're going to die eventually. So that's what is actually happening, and that's what is limiting these microorganisms going to those spaces. Yes. I mean, so there should be a low temperature cutoff to be metabolically active. But what happens at low temperature is that you could go into a freezing state where you could not be metabolically active, but just sustain your viability forever. So that's kind of like, it gets a little bit complicated. And I first thought high temperature would be more complicated, but turns out colder is much more complicated. So I mean, I would love to work on that, but I just don't know how. So if you have ideas, I would love to. Yeah. Yes. Yeah, perfect question. I forgot to mention that. So how this can inform us about the LUCA debate, whether it was low temperature or high temperature, is that the ultimate conclusion I'm having is it doesn't matter. LUCA was simple enough to be at mesophilic condition, thermophilic condition, and hyperthermophilic condition. And that's why probably maybe we have this confusion of whether it was a low temperature or a high temperature. Because if you are simple enough, as I showed you in the first plot, you could make that low complexity across all the temperature regimes. So that's where, I mean, it gets complicated to really, really pinpoint whether LUCA was thermophilic or hyperthermophilic. But I think it really doesn't matter to us as long as it's simple. So what we could inform is that LUCA has to be simple. unless we find a definite proof that it's mesophilic and it was more complex. You had a question? So indeed, so all these parameters that I said, these are all temperature-based parameters, and they don't have to be temperature-based parameters. If you could figure out how these, catabolic energy at the rate at which you get and the maintenance cost would vary with regards to pressure. So what I'm trying to do is I'm trying to estimate this in lab, where I culture these microorganisms at different temperatures, and I see how that the exponential curve actually increases. But if you're, say, interested in pressure, you could do the same, and you could have a plot where it's like, okay, this is how the maintenance cost varies with pressure, or say pH. And now you know that, and you can, again, use how those chemical reactions that get the energy in behave with regards to pressure and pH conditions. And that is going to inform you about how you might find a similar plot for pressure. So that's not what I'm working on, but you can definitely do that with these kind of models.
We're gonna move on. Thank Jay again. Okay, our next speaker will be Sarah Gonzalez-Henao, talking about biopolymers and serpentinization, associated extreme environments, and their significance for the habitability of microorganisms in extraterrestrial systems. Take it away, Sarah.
Good morning. Good morning, everyone. So my name is Sarah Gonzalez. I'm a third year. Okay. Hello. Yeah. So my name is Sarah Gonzalez. I'm a first year PhD student at Michigan State University. And today I will be talking about biopolymers in serpentinizing systems and the implications for habitability and life detection. So among the extreme environments on Earth, one of the most intriguing are serpentinizing environments, which are a product of water-rock reactions. We have the serpentinization reaction, which consists on the reaction of olivine and pyroxene minerals with water, which produces the serpentine mineral. And as a byproduct, we have hydrogen and methane, when we also have heat. And this reaction produces high pH conditions. And on Earth, we have marine serpentinization, such as in the Lost City hydrothermal vents. But we also have terrestrial serpentinization or continental serpentinization, such as in the Santa Elena ophiolite. And today, I'm going to focus on these two serpentinizing systems, the Lost City and Santa Elena. So for Santa Elena ophiolite, as I said, it is a terrestrial serpentinization site located in the northwestern Pacific coast of Costa Rica. And it is quite intriguing because it has a strongly contrasting wet and dry seasons that makes Santa Elena a unique hydrogeological setting, where the water-rock reactions are enhanced by the large storm events. And there are also hyperalkaline springs being produced of about pH 11 point high, so it's quite high. And interestingly, Santa Elena ophiolite is a Mars analog, considering that within the Niliphosaur region of Mars, the Mars Reconnaissance Orbiter detected serpentine minerals. And there are a lot of mineralogical similarities between Santa Elena and the Niliphosaur region of Mars. So it is a potential analog that can be studied. On the other hand, we have the Lost City hydrothermal field, which is a deep sea serpentinizing environment located in the mid-Atlantic ridge. And it has high temperature conditions, is metal poor, it is highly alkaline, a pH of around from 9 to 11, and there is a low biomass but high species diversity. And there are actively venting carbonate chimneys in the Lost City. And interestingly, the Lost City is an analog of ocean worlds, specifically Europa and Enceladus. And considering that there might be hydrothermal activity in these two moons that can provide habitable conditions for life, such as liquid water, but also chemical energy for the metabolism of microbes. So within the Lost City, but also in Santa Elena, there are active microbial communities growing. And a very intriguing question is how these microbial communities are thriving at such high pH and high temperatures. So there are different mechanisms that microbes can use to survive, and one of them are biofilms. So this is a protective layer that microbes secrete in order to protect themselves, and they create a microenvironment. So biofilms in general are secretions that facilitate attachment to surfaces. In this case, could be mineral phases, and that lead to the formation of microbial communities, microbial biofilm communities. And they have many functions, including drought protection, signaling, trap of nutrients, among others. And these biofilms are composed of extracellular polymeric substances, or EPS, that are comprised of 5 different biomolecules, the exopolysaccharides, which are the main component. We also have proteins, extracellular enzymes, eDNA, and extracellular vesicles. And biofilms are of astrobiological relevance, considering that they have implications for the habitability of microbes in extreme environments, but also in extraterrestrial systems. And they can also provide new biosignatures to detect life. Since some of these biomolecules are highly resistant to degradation, and they can interact with mineral phases that aid in its preservation. So these biofilms have not been studied in depth in Santa Elena and the Lost City. So why studying biofilms in serpentinizing associated extreme environments is relevant? Because they can inform us about the habitability on Mars, Europa, and Enceladus. They can inform about the strategies that microbes use to cope with the high pH stress. And also they can offer new biosignatures to detect life. So we used some sequencing data, specifically metagenomic data, to analyze biofilms specifically from Lost City and Santa Elena. And therefore, we were interested in exploring the potential of EPS biosynthesis by bacteria in Santa Elena, but also in the Lost City. We're using a metagenomic approach. So specifically, we focus on the exopolysaccharides, which, remember, are the main component of the biofilms. And so we look at the biosynthetic gene clusters that encoded for these exopolysaccharides in the metagenomes of these two environments. And in second place, we wanted to know what type of microbes were producing these exopolysaccharides using metagenome assembled genomes, which are genomes reconstructed from DNA of microbial communities. And in third place, we analyzed the relationship between minerals and the exopolysaccharide genes that we found in these metagenomes. This one was specifically for the Lost City. And we analyzed two carbonate minerals, brucite and aragonite, which are common minerals in the carbonate chimneys of the Lost City. And so the samples collected from Santa Elena are from two hyperalkaline springs, which had a pH of around 11.5, so that's quite high. And for the Lost City, the samples were collected by Dr. Osama Alian during a deep sea expedition to the Lost City in 2018. So what did we find in these metagenomes? So for the Lost City, we found a wide diversity of exopolysaccharides being produced. So we found 10 different types. And the Sphingin exopolysaccharide had the highest abundance. For the Lost City, we found similar trends. So we found a high diversity of EPS types, around 11 types. And again, the Sphingin was the most abundant. So interestingly, the Sphingin is actually a family of exopolysaccharides that are anionic EPS that can produce agar-like structures. They have highly conserved biosynthetic genes, but most importantly, they have high stability over wide pH ranges and wide temperatures. And this is the main reason of why of the abundance of these EPS under these high pH and high temperature environments. In second place, we wanted to know what type of, what microbes were producing these EPS. And so here I have the results for Santa Elena. Here you can see the order and the different types of EPS being produced. And we found in general a high number of exopolysaccharides produced by the different microorganisms, where Santa Moladales had the highest EPS had the highest diversity of EPS types, and it is a well-known alkalophilic order. And we also find that Burkholderia was enriched in the Sphingin genes. And these are the results for the Lost City. Again, we see this is the order and the different types of EPS. We found a high diversity of EPS being produced by the different bacteria. And in general, the Methylococales had the highest diversity, followed by Pseudomonadales. And this diversity of exopolysaccharides being produced highlights the critical role for establishing the biofilms and conditioning the environment for further colonization of microbes that allows them to thrive at such high pHs, high temperatures. And so In third place, we did some correlations of these exopolysaccharide genes that we find with some mineral data, specifically brucite and aragonite. And in this table, you can see the most significant correlations for the exopolysaccharides, which were for the sphinxin family, but also for the polyenacetic glucosamine. And we found very interesting trends. Like in general, we found positive correlations with brucite, but negative correlations with aragonite. So why is this? Brucite is a magnesium hydroxide mineral that can act as an absorbent of organic compounds. And it is stable at high temperatures. And remember that these are conditions that we see in the lost city. So this can help explain this positive relationship. But also, for the sphinxin family, we have high stability over a wide range of temperatures. So brucite can act as a mineral phase for the formation of biofilms. And these findings are supported by this confocal laser scanning microscopy analysis that Dr. Osama Alian did. So here you can see a section of the hydrothermal chimney. And in cyan, you can see the brucite, and in red, is the aragonite. And when he stained with DAPI, which is a stain for the nucleic acids, here you can see bacteria, conglomerates of bacteria of biofilms associated directly with the brucite. So coming back to the table for the aragonite, where we saw negative relationships, this can be explained by the unstable nature of aragonite, because this is a mineral that tends to convert over time to calcite, which is the final stable form. And so to conclude, bacteria in Santa Elena and in the lost city have the capacity of synthesizing different exopolysaccharide types. And this abundance and diversity of EPS types has strong implications for astrobiology, specifically in terms of the habitability that they can provide to microorganisms, but also can provide potential biosignatures for detecting life. And Santa Elena and the lost city, which are a terrestrial and marine serpentinizing system, can serve as a potential analogs for Mars, but also water worlds, such as Enceladus and Europa, and are key sites for astrobiological research to study habitability and life detection. And future directions. So for my thesis research, I'm going to do a holistic study on biofilms from Santa Elena ophiolite. We recently did a sampling trip to Santa Elena during the dry season. And we found two very interesting hyperalkaline springs where we collected biofilm samples. So this is one of them. It had a pH of around 11.6. And these other hyperalkaline springs was quite intriguing because it was located in a small steep. And at the bottom, where the river was flowing, the pH was 8.86. And at the top was 11.25. So there was a pH gradient within less than a meter. So it was like quite relevant for like studying microbes and habitability conditions. And so what do we want to do with these biofilms? We want to analyze, do metagenomic analysis, metatranscriptomics. We want to characterize the diverse EPS components, not just the exopolysaccharides, through different approaches, so microscopy, biochemical, and spectroscopic techniques. And characterize the EPS mineral interactions, as it's highly relevant for biosignatures preservation. And we hopefully want to do another field sampling trip during the wet season to compare like both in dry season and like the dynamics of it, of the biofilms. And with that, I would like to thank my advisor, Dr. Matt Schrenk and the Schrenk Lab, Dr. Ricardo Sanchez Murillo, William Brazelton, and Dr. Osama Alian. Thank you very much.
We have 5 minutes for questions.
Hi, great talk. I'm wondering, at the field sites, how do you distinguish, or do you distinguish between biofilms you're collecting and like carbonate mineralization, or do you just assume this is all a biofilm and collect it all?
Well, usually people think of biofilms as thick, like secretions, like you see here, like here. But biofilms could also be like conglomerates of cells that are not being seen. So we collected directly like carbonate minerals from these springs. So you can see there were a lot, like at the bottom of this rock, we found a lot of carbonates, which we collected and preserved. And we want to do with that, we want to stain them with DAPI to get to see like conglomerates of cells, and where we can tell that there might be a biofilm. So, so we found like, we took these carbonate minerals, but also we took like these kinds of slimy biofilms. So there are different forms in which you can find them.
Thanks for the talk. How abundant are the EPS producing mags overall compared to the? How abundant are the mags specifically found to contain the EPS genes?
Like
Relative abundance, your sample for those mags.
For instance, for the Lost City, we found, so for the Lost City, we had like more samples, like around 10 samples. So we were able to construct more metagenome assembled genomes. And in the relative abundance of these, so we did a filter of the like high quality mags, and like the relative abundance was like higher than, like, I would say 60%. So it was like quite for the Lost City, which we had like more samples. In contrast, for Santa Elena, we had just 2 metagenomes that were collected previously. So we were able to build, like, you see more like lack of information compared to the Lost City. So we were not able to filter a lot of high quality mags, and the like abundance was less than like 50%, I will say. So that's why we want to focus on like collecting more biofilm samples from Santa Elena, since we don't have like a lot of information. And I took some previous data to like look into biofilms.
Thank you very much.
At your terrestrial ophiolite, are you seeing in your thicker biofilms like laminations that might be stromatolitic, or like at least a trapping and binding of pearl aggregate or anything? Just like the proto or a stromatolitic feature in that thing?
No, we're not seeing like stromatolite like formations. So we're actually seeing more like very like carbonate, soft carbonates mixed with water, but not like rocky formations in the hyperalkaline springs. But for this one, we actually like found layers of carbonates that were actively precipitating carbonates. So that was kind of interesting. So we also took samples of that, but we mostly found these types of like carbonates mixed with water on the bottom of the springs. Thank you.
All right, our last talk in this session will be from Sylvia Knupp, and her talk is titled Identifying Translationally Active Microbes in the Guaymas Basin Deep Biosphere. So take it away, Sylvia. Also, this is a really cool video, how to start this off.
Thanks. And is that working? Yes? Okay. Not yet?
Is it good? Yeah.
Good? All right, sweet. Hi, thank you for the introduction. All right, what you're seeing here is a submarine landing on the bottom of the seafloor at Guaymas Basin with a payload to collect samples at this site. So this is going to be my study site for today. In our search for life on other planets, one of the main focuses might be life in the subsurface of planets and moons. The reason being, these environments are probably more protected from space hazards like UV radiation, meteor strikes, etc. And these environments might also harbor more conditions that are like life as we know it. For instance, on Enceladus, the subsurface oceans are liquid water. They have hydrothermal vent systems, which might heat up the water, and they also have organic molecules present. However, we don't have good ways to study these ecosystems yet, and so we need to develop these methods before we have any chance of figuring out if life exists here. To do this, we can use subsurface environments on Earth as an analog site. So the deep biosphere of Earth is defined as any organisms living below 1 meter below the surface of the Earth. So anything deeper than that. All the organisms and rocks, all that sort of stuff. This environment is much more extensive than we have thought previously. By some estimates, 65% of all microbes live in the subsurface. But these environments are really, really difficult to study. For one, cell abundances are very low. As you can see here, as we get from the surface, where our cell concentrations are like 10 to the 8th cells per cubic centimeters, even 100 meters below the surface, this concentration drops to less than 100 cells per square centimeter. So this makes it incredibly difficult to study, not to mention that a lot of these organisms are dormant or not very active, and so that makes a lot of our traditional microbiological techniques really difficult to execute here. So for instance, our culturing methods do not work as effectively. So we need other methods in order to study these microbes in this environment. My lab uses this method called BondCat FACS, which is bioorthogonal, non-canonical amino acid tagging tied with fluorescence-activated cell sorting. That's like a really big mouthful, but the important part is that we're trying to identify translationally active microbes by tagging those proteins that are being newly made. So the way we do this is with a synthetic amino acid called HPG, which is a substitute for methionine. So we can incorporate this into cells that are making new proteins, extract these cells from the sediments, and then attach a fluorophore to this HPG molecule using click chemistry. And so now we can have fluorescent cells that are the ones that are making new proteins. We can use this to our advantage to put into a fluorescence-activated cell sorter, so we can sort out these active cells, and then we can take these cells and we can sequence their DNA in order to determine the identity of these cells. So my goal in this is to apply this to the deep biosphere of Guaymas Basin, and so any samples below the surface. And I'm particularly interested in the activity of all 3 domains of life: bacteria, archaea, and eukaryota, and seeing if all 3 domains of life are active. In order to do this, the samples need to be collected on the IODP 385 cruise on the JOIDES resolution. They took gigantic cores down to 150 meters below the seafloor for microbiological analysis. We took these samples, incubated with HPG, attached the fluorophore. Now we have active cells, and that's what we're searching for, is this active fraction of cells. So our lab has developed this BondCat FACS methodology, and it works really, really amazingly for both bacteria and archaea, but it has never been tested on eukaryotes before. So we were particularly interested in fungi, and so we needed to test if we could even get fungi to incorporate HPG into their cells. We have 2 marine fungi species. This is Rhodotorula, which is a marine yeast, Cataphora, which is a marine filamentous fungi, and we needed to test if we could label them, which we can. Woohoo! So the Cy5, which is the red dye, is our click-stained dye. So all the cells in this sample, in this synthetic environment that are red, are ones that click-stained. And so we've got these the long filamentous cells that you can see around, that's the Cataphora. These small, like circular cells, that's the Rhodotorula. And if you look closely, you can see these little rod-shaped cells, which are E. coli. And so another problem that we had is in cell sorting, we need to be able to sort these gigantic cells alongside the tiny prokaryotes that are probably the majority of our sample. And so we needed to make this synthetic environment in order to see if we could do this at all. And the answer is, yeah, it works pretty well. We can sort them all at the same time. So after doing this, we can apply this to my study site, which is Guaymas Basin. So Guaymas Basin is a fascinating site. It's a hydrothermal site in the Gulf of California, near the Pacific Ocean. It's got 5 rivers that flow into it, depositing tons of sediment. It's also highly productive. It's close to the equator. It's got tons of photosynthetic organisms. And so what happens in this environment is as the sediment and as the sediment falls, as these photosynthetic organisms die, they all fall to the seafloor as what we call marine snow. And they interact with the hydrothermal vent environments there. And they thermally alter this organic matter, and it makes for a really cool bed of chemistry for life. So here's a hydrothermal vent from Guaymas Basin. These vents are producing primarily methane, which is really unique for a vent site. This is because it's thermally altering the carbon that's falling down as marine snow. They're surrounded by these microbial mats, which are Beggiatoa, that are kind of populating the whole seafloor here. So it's a really rich, really hot, really active environment on the surface. When you go below the surface, it looks quite a bit different. It's kind of this boring mud. Even 1 meter below the seafloor, you can no longer see the signs of the microbial mats on top. All the way down the sediment column, it's just kind of this brown, oily, very smelly mud. But we think that even in this bottom section, which has cell concentrations less than 100 cells per centimeter, there are active microbes. So we need to identify this active fraction of microbes. We are using cell sorting to do this. Basically, we send our sample, which theoretically has click-stained cells, into the cell sorter, and we get this little cloud of events. And we have fluorescence on this y-axis. We draw a gate around where we think the positive cells will be based on a negative control sample. And this is 154 meters below the seafloor. Lo and behold, we get an active fraction of cells. So that's what we're calling this, the active fraction. We extract the DNA here, and we get our 16S rRNA gene amplicon sequencing data. This is a big chart, so I'm gonna walk through the first section real quick. On the left, we have our kind of code here. The pre-sort is all of the cells in the community, whether they were labeled or not. So that's the entire community. The sorted cells are the active fraction of cells that we sorted out. And we have classes of organisms sorted by abundance. And so if the circle is larger and more yellow, they're more abundant in that particular sample. So this is our first sample, which is just 1 meter below the seafloor. And we go all the way down to 154 meters below the seafloor. These are incrementally down. And we find tons of microbes. It's awesome. One of the ones that sticks out the most is this gamma proteobacteria. They're super common in the entire sample. These are likely heterotrophs that are consuming the organic matter that falls down from the surface. We also have really large populations of alpha proteobacteria, bacilli, and dinococci. These are also heterotrophs. They're likely consuming organic acids, some polysaccharides, maybe some aromatic compounds. And the really cool thing about this is that, especially with the dinococci, you can see those larger circles for the sorted fraction of cells, which means that not only are these guys present, they're also actively producing new proteins in this environment, which means that they're likely consuming these things that we predict from their genomes. Diving into the gamma proteobacteria a bit further, down to the genus level, we can see some very interesting trends for the pre-sort and sorted samples. So this Halomonas is pretty common in a lot of samples, but you'll notice that the circles are much larger in these sorted samples. And so these guys are likely more active than you would assume if you were just doing regular abundance studies. So we also see the same trend for this Acinetobacter group. One of the more interesting things is that down in this bottom section, which is the lowest depth, 154 meters below the seafloor, they're almost not present in the overall community. You're like barely detecting it. There's one dot there. But when we looked at just the active fraction, they're super abundant. This is indicative that they're making up a huge section of the active population, even though they're not very common in the overall community. These guys are likely way more important than we would predict by genetic data. We see the reverse trend for this Parabricoldaria, where they look super common in a lot of the samples, but then when you go and look at the sorted sections, the active sections, they're not very common. These guys, probably a lot of DNA makes it here, but they're not really doing anything. Or if they're doing stuff, it's like at a very low level. So they're likely less important than the rest of the environment. Most of these samples are bacteria, but of course we've got some archaea here in the other corner, and these guys are also looking very interesting from these samples. We have two classes of the Asgard archaea, which are archaea closely related to eukaryotes, and they're present in mainly the surface sediments. These guys are heterotroph generalists. They can consume complex sugars and peptides and potentially some acetate. And what's interesting about them in this environment is that they are in just the surface sediments. So theoretically, based on what we've seen from the bacteria, all of their carbon substrates should be at the bottom, but for some reason they're not there. So something that they need must be missing at these deeper depths. Additionally, we have two classes of methanogens that pop up, the Methanomethyleceae and the Methanosarcina. These guys can consume methylated compounds and produce methane. So if you remember earlier, I told you that a bunch of methane is produced by the hydrothermal vents. There's also a biological source of methane now. So organisms that are consuming methane in this environment could be getting it from the hydrothermal vents, but now they could also be getting it from these deeper samples of the methanogens as well. We're still awaiting our fungi sequences, but once we get them, we'll be able to see kind of the comparison of what they are consuming compared to what the bacteria and archaea are doing. This is one of our samples of the pre-sorted samples, so the whole community, but labeled. And so you can see tons of those red little dots. That's our click stain. Those are our active cells. There's quite a few of them, even really deep. So we have a ton of these little cells, which we assume are prokaryotes, but there are also some larger ones, like this big circle up top, which could be fungi, although we don't know for sure until we get the sequencing data back. Based on previous results, we see fungi at these depths from culturing efforts, and they're heterotrophs consuming very complex matter like cellulose. So it's possible that they are consuming these really complex organic matter, and that's feeding into the bacteria and archaea. It could also be possible that these guys are competing with the archaea and bacteria, and we'll be finding out soon enough. So we've determined that BONCAT-FACS is a viable method to detect active organisms in the deep biosphere from all three domains of life. We've found that archaea and bacteria are both active in the deep biosphere. We've also determined that heterotrophs are kind of dominating this environment, even at deeper depths. And so it's likely that this organic matter is ending up in these deeper samples much more than we previously assumed that they would be. And most importantly, life exists deep underneath the surface of the Earth. These guys are consuming everything from the surface that falls down into these sediment layers, and it's likely that life on other bodies could be doing the same thing, being protected from UV, from cold temperatures, but still benefiting from surface chemistry. And so we need to really further explore the deep biosphere on Earth in order to determine what the actual limits of life are as we know them. Thank you, guys. I want to thank my lab, the Hatzenpickler Lab at Montana State University, specifically Dr. Andrew Montgomery, who I've worked with me on this entire project, and our collaborators, Dr. Ginny Edgecombe at Woods Hole Oceanographic Institute. She is the fungi expert on this, and Dr. Andreas Teskey, who was the lead scientist on the cruise. With that, I'll take any questions.
We've got about 2 minutes for questions.
Hi, thank you for your talk.
Thank you.
So I was wondering for your incubation time for the Boncat, how long were you incubating these cells? And are you seeing that you need to incubate the cells longer than you would for someone you or something?
Yeah, that's a really good question. So we didn't know how long it was going to take, and so the cells were incubated for 2 days, 3 days, 7 days, and 14 days. We ended up finding that 7 days was plenty, but 3 days we weren't really seeing many active microbes. And so they're definitely way slower in producing new proteins than a surface sample, which I've done work on where I can incubate in like a day or two.
Hi, interesting talk. I'm Abby Deering from Georgia Tech. The question I had was about the fluorescent tag that you're actually using, and if there's any preference given to using that or not using that compared to native amino acids and untagged amino acids, and how you're sure that it's actually integrating into the system rather than being ignored by the metabolism of the system.
Yeah. That's a great question. So let's see if I can back here. So the way that we are basically doing this reaction is it's a click chemistry reaction. And so there is an azide group on the HPG molecule that can only click to bring their, oh my gosh, I've forgotten all the names. It can only do that by a copper catalyzed reaction, which we do after we take the cells out. And so that is kind of an exclusive reaction, and you're not likely to find those like end pieces in a lot of the cells. And so that's how we kind of guarantee that those are tagging the exact proteins. Sure.
Can you give Sylvia another round of applause? And if you have any questions for any of the presenters in this session, feel free to ask them over the break. So we've got a 20-minute break. For the presenters in the next session, could you possibly come up here just to make sure that we could get your talk uploaded? I put on the conference chatter channel like a form. If you had problems uploading, just come see me. And for future questions, can you all please say your name and what university you're from before you ask your question? All right, feel free to come back at 10:40.
watching the Weather Channel, your most accurate and dependable source of weather information 24 hours a day.
Hey, good evening. You're watching the Weather Channel here on a Friday evening. Sequentialist Janetta Jones, and as we look out there, Janetta, look for
All right, our next talk in this session will be Peter Winslow, and he'll be talking about structural insights into the origin of life through model primordial proteins. Take it away, Peter.
Thank you. Everyone hear me? Is this on? Perfect. All right, so thank you so much for giving me this opportunity to speak about what I've been working on for the last few years. So my lab is primarily a protein engineering lab, but we have heavy interest in the origins of life. We're trying to look at the origin of life through a protein biochemistry lens. And when we're looking at the origin of life through that lens, we really want to know how the first proteins may have emerged and how they might have acted. So in this way, I like to compare my work in our lab to a sort of weird version of Jurassic Park. So I like to call it like, oh, we're in the Primordial Park, right? Where we're trying to almost bring back to life very ancient prebiotic versions of proteins that might have existed. So in order to do this, we need to think about the timeline of protein evolution, where the earliest would have been abiotic synthesis of primordial amino acids. So this is really all chemistry. This has been experimentally shown to be possible. We have very famous experiments such as Miller-Urey, and then we have analyses of meteorites that show that you can get amino acid synthesis without any biotic intervention. These would eventually randomly polymerize into very small, likely completely unfunctional polypeptides, which at some point transition into larger functional proteins, likely composed of very primordial sets of amino acids. And then this transitions over hundreds of millions of years into the modern proteins that we see today with 20 proteinogenic amino acids. And this has sort of been what life has used since Luca 3.5 billion years ago. What I'm particularly interested in is this period of time where we're going from random, unfunctional polypeptides into functional proteins that would have given some sort of fitness advantage to whatever early form of life existed at the time. In order to do this, we need to think about what amino acids these early primordial proteins might have been composed of. So we use the Trifonov consensus chronology, which is an attempt to order the amino acid incorporation into biology. So on the earliest side of things, we have very simple amino acids such as glycine, and then on the most modern side, we have very bulky, sort of metabolically complex amino acids. With this timeline, we can then make arbitrary groupings of these amino acids into what we call alphabets, meaning there's different subsets of what amino acid the protein can be made out of, where we have the most complex at 20, which is essentially modern proteins. Then we get a little bit less complex with 16. In my opinion, the most interesting, which is the 9 amino acid alphabet. All of these amino acids are able to be synthesized in the Miller-Urey experiment. And then a sort of extreme example with the 5 amino acid alphabet with only 5 extremely basic, not chemically basic, but extremely simple amino acids. With these alphabets, we then chemically constructed 10 trillion random proteins, so 80 amino acids long, but only consisting of amino acids in each of these 4 alphabets. So now that we have actual proteins made that should be decent models for what might have existed at that origin point, we can start to ask questions about what we think these proteins might have been doing. So the main questions we're really interested in is, can you get biological function from these primordial alphabets? If they are functional, is there any relationship between the structures we see and the alphabets they come from? And then, similarly, are those structures similar to what we see in modern proteins today? In order to answer these questions, we need to actually get functional proteins. So we selected for proteins that bind to ATP due to its ubiquitous role as an energy currency in life. This was successful. This was done by Matilda Newton and Kunwal Lee previously. I'm only showing here the results from 9 amino acid library for the sake of time, but we found binders from every library, and then we kinetically characterize a handful of them. And what you see here, based off this binding assay curve, is that 3 of the variants from the 9 amino acid library, again meaning it's only consisting out of those 9 amino acids that could be found in the Urey-Miller experiment, have binding affinity to ATP that is functionally indistinguishable from modern proteins. So this is a huge deal. It means that from sort of random sequence space, you can find proteins that behave, at least on a surface level, very similar to things that have evolved over billions of years. But the big question is, how do they do this? And how indistinguishable is the structure and method of this function? So in order to sort of justify this, why do we care so much about the structure? So I need to introduce sequence space. If you're not familiar with this term, think about every possible combination of amino acids as a huge space that you can sample out of. And on the sort of y-axis, or z-axis, I guess, you have how good at some given quality that protein is. So in our case, binding to ATP. When we look at these protein structures, there's really two possibilities. Either they look very similar to modern proteins, or they look not like modern. This is interesting because if they look similar, that implies that life has maybe sampled enough of the sequence space that it's found the optimal way to bind ATP in our case. So you can imagine at some point there's an origin of life event, and then evolution takes hold and it slowly increases fitness until we reach this point where life really doesn't have much improvement that it can make. Alternatively, if we see that it's unlike modern proteins, that might imply that life is a frozen accident, meaning that some origin point occurred and it reached a peak, but due to the fact that there's these detrimental valleys around it, it can't get out. And therefore, there's a lot of unexplored, potentially really interesting biochemistry. My hypothesis is that we're more likely to find these novel binding mechanisms that imply a frozen accident due to the fact that most of these alphabets are extremely limited in the amino acids that they use. So for some context, this is a previously selected ATP binder from a random sequence, but it contains all 20 amino acids. This was done by Tony Keefe in 2001. And at a glance, you see the structure, and it doesn't look anything super indistinguishable from modern proteins. However, it is somewhat novel. The distinct components that bind to zinc and the ATP are reminiscent of what we see in today's proteins. However, the combination of them is novel. So this isn't really an answer to either of those scenarios. It's sort of a merger of the two. So when we look at how it binds to ATP specifically, it uses positively charged residues to bind to the phosphate groups, and then aromatic pi stacking with the adenosine moiety. So this is very similar to what we see in modern ATP binders. The way that they were able to make these observations was through X-ray crystallography of the protein structure. And we would like that to be possible for all of these proteins that we're interested in. We have over 20, though. And X-ray crystallography is somewhat low throughput. So in the meantime, while we're trying to get structural data, we can use computer modeling to try to give us a better idea of what we're working with in terms of structure. So I'm sure a lot of you have heard of AlphaFold. It's sort of a groundbreaking protein structure modeling tool. So AlphaFold 2 was the big deal for the last few years. And the important thing to know is that it largely relies on what's called multiple sequence alignments, meaning you give it a sequence of your protein and it compares it to many sequences in a database. And anytime there's something that it thinks is similar, it'll look at the known proteins and say, what does this look like? And try to fit that into the sequence that you give it. The problem with that is our proteins are not related to anything that has ever existed, and their sequences are very strange. So AlphaFold sort of struggled with this. You can see on the bottom is the confidence rating, where the more orange is less confident, and these structures are very unconfident. And if you know anything about protein folding, they don't look very well folded. So this was a little disappointing, but not a huge deal. We didn't really expect anything groundbreaking from this. However, about a month ago, AlphaFold 3 came out. And part of the change from 2 to 3 was that they claimed to have moved a little bit away from that MSA, that multiple sequence alignment. So I was really excited. The day it came out, I plugged all these sequences into it to see if it got any better. And unfortunately, they didn't really get any better. In fact, 9C looks worse, even though it's a little bit more confident. I would say 9D is a little bit better looking, but again, not confident enough to really believe that it thinks it knows what it's doing at all. However, the real cool thing about AlphaFold 3 is that it allows you to do predictions with ligands, meaning that ATP molecule that these proteins are known to bind to. When we introduce ATP and metal ions, suddenly the predictions become much more confident and much more realistic looking. So these are completely in the range at which AlphaFold 3 says that it thinks that this is what the protein actually looks like. So this was super exciting. By no means do I trust this yet. These need to be experimentally validated, but still, it was really exciting seeing all this blue pop up for the first time. So hypothetically, let's look at 9C. If this was the structure, if AlphaFold 3 is correct, what would we look at this and say about that protein sequence space that I was talking about earlier? So if we zoom in on this molecule, we can see a surface of the protein, and you actually see a sort of realistic-looking binding pocket that ATP could fit into. And zooming into that binding pocket, we see a network of hydrogen bonding that is somewhat realistic and not seen in modern ATP binders. Additionally, we can compare it using a tool like FoldSeq, which is a protein structure comparison search tool, to modern protein structures, and try to determine on a global level, is the structure similar to what we see in modern proteins? And based off of this sort of example run, we see blue is the 9C AlphaFold 3 structure, yellow is a motor domain, and based off of these scores, this is a poor fit. So based off of the fact that the hydrogen bonding of the ATP that we see is not similar to what modern ATP binding proteins use, and the fact that we're unable to find any largely similar protein structures, if this was truly the structure that we find, we would make the case that, we'd make the claim that this sort of makes it more likely that life is that frozen accident scenario, which I think is really exciting, meaning there's a lot of interesting protein biochemistry that's out there to be studied. Additionally, what's I think really cool about AlphaFold 3 is it gives us an opportunity to prove people wrong. If we show that the structures of our proteins are not what AlphaFold 3 predicts, then we potentially have a large gap in knowledge to fill that I think is really interesting. So this is some very preliminary structural data that we have obtained thus far. So on the left, I'm showing a circular dichroism spectra. This allows you to determine secondary structural components of proteins, which are these beta sheets and alpha helices shown in that purple and blue. And so based off of this, it seems like 9C, that protein I was showing earlier, is about 43% alpha helical. Based off the AlphaFold prediction, it should be less than 20. So already there's some discrepancy there. Additionally, we can look at HSQC nuclear magnetic resonancy, and this spectra gives you an idea of the sort of global fold of the protein. And based off this, it might actually entail that this protein is largely disordered, which is also interesting by itself, meaning it's very sort of flexible and transient in its structure. So either of these scenarios already contradict AlphaFold, although they might not agree with each other. Again, this is preliminary, and we're sort of ironing these out. So with that, I'd like to just summarize. We have ATP binders from primordial alphabets that are functionally indistinguishable from modern proteins. Structural modeling has been interesting, but we need to experimentally verify it. So for now, it's questionable. So we're collecting structural data to do this, but that is a huge work in progress. But hopefully soon, tentatively within the next year, we'll get a lot of interesting structural data. So with that, I'd like to sort of zoom out. And I know that there's a very wide diversity of fields here. But when you look up in the sky, you see all these stars, you think of there's just like a near infinite number of worlds that might harbor weird life. And it's just sort of mind-boggling to think about. Protein sequence space is the exact same way. And so in another sort of nerdy sci-fi analogy, I like to think of our lab as a sort of Star Trek ship where we're exploring protein sequence space where no one has gone before. So if you remember nothing else, remember that we're exploring really weird proteins and might discover stuff that no one has ever seen before. So with that, I'd like to thank everyone, particularly from my lab. Kunwa Lee laid a lot of the groundwork for this work, and then my undergrads who really helped me do a lot of this, and then our funding, and everyone else who's helped me with this. So thank you.
All right, looks like we've got a couple questions.
Hi, Abby Deering, Georgia Tech. Interesting talk. I was wondering, and I'm not a prebiotic chemist, so maybe this is obvious, but why are you looking at ATP binding if you're trying to mimic super simple early solutions? Like, isn't ATP kind of complex for these early proteins to be binding to?
We chose ATP mostly due to the fact that it's seen in so much life today, and it has a lot of things that we can compare to. It's not like a perfect system that we're saying this is what life looked like when it started. It's a very broad model system. So we're not really too attached to the idea, is the ligand super prebiotically viable? Although I think ATP has been shown to be somewhat prebiotically viable, especially when you're looking at like RNA world. You need something that can be the building block of those RNAs.
Thanks.
Yeah.
Hi, Taylor Polka, Notre Dame. Nice talk. I do a lot of work with like RNA world type stuff, so I know a lot about like the plausibility of like RNA self-polymerization, but I don't know much about like what like that looks like for proteins and amino acids in a prebiotic environment. So if you know anything about that, I would be very interested.
So protein, specifically, there's not really protein self-polymerization right now. It's all polypeptide.
Okay.
The longest that I know has been shown is about a 30 mer. So ours are 80 amino acids long, so quite longer than that. The reason we chose that length was because it's short enough that it's feasible to work with, because we're working with tens of trillions of random sequences. But it's long enough where you can get folds that might look similar to what we see in modern proteins. Yeah.
Hi, I'm Costa from UW-Madison. Peter, great talk. I had a question, like a couple of similar questions. So what I was thinking is, have you looked at the current ATP binders and how similar are the binding domains within themselves?
Yeah.
And the second question I had on follow-up of that is like, again, how different is the binding domain, let's say the one you found from AF3, is it from there? And the ones that you see in the extant organisms, let's say, that are bind to ATP, like how heavily is it occupied by the primordial amino acids? Like, do you also see like, I don't know, the later ones in there? So how much would it be affected?
So here's some examples of natural ATP binders. These all share one common fold or motif. So motif is a sort of a common way that a protein, in this case, binds to something. So this is called the Walker P-loop motif. This is really the most common way that modern proteins bind to ATP. There's a few others like Rosman motif. And you can see that it always has this beta strand and an alpha helix, and this little loop is always glycine rich, and there's a lysine residue, which actually interacts with that phosphate group. And so that's a positively charged residue that occurs in our timeline in the 16 amino acid library, which means that the 5 and 9 amino acid proteins absolutely cannot have this motif, just necessarily. Additionally, when we look at like the fold of what the alpha fold thought 9C looked like, it does not display this sort of beta strand alpha helix. So we've looked into that a little bit. One of my colleagues, Sam, is actually looking into that a little bit, trying to fish out if there's any of these motifs from the binders that we selected.
Hi, I'm Alex Deansrow from Georgia Tech. Good talk. I'm curious if there are a lot of hydrophobic residues in that protein you found that may have an intrinsically disordered structure to it, because I was just thinking that maybe that might be why sequences like it aren't found in life, because if you have a bunch of exposed hydrophobic stuff, then it will be prone to aggregating.
Yeah, that's definitely a possibility. The good news is, at least at the concentrations we're working with, we don't see any aggregation, but there's a ton of valine, which is hydrophobic. So that's really that's the big question, is we're trying to see what properties do these have. Yeah.
Thank you.
Yeah.
Give another round of applause for Peter. And I know a couple other people had questions. Just feel free to ask during lunch after this next talk.
Can you all hear me? Sweet.
Yes. Give me 1 second. All right. Our next speaker, last speaker in this session, will be Haley Brown, talking about elucidating planetary scenarios that may lead to an origin of life in warm little ponds. Take it away, Haley.
Hello. Like I was already introduced, I'm Haley Brown, and I'm a first year at Purdue University, and I am very excited to talk about warm little ponds and planetary features. Oh, I did not get the clicker. There we go. Cool. So in the search for life, we all talk about habitability like a ton. We have the habitable zone, which I have pictured here, where we are really interested in looking for life, and it's just a term that we all talk about a lot. But what it boils down to is a location's ability to maintain liquid water and/or its ability to sustain Earth-like life if it was introduced. And as we're getting more and more data and exoplanet stuff, which I'm super excited about, we need to think about narrowing our criteria to actually highlight planets we think could have the highest possibility of being inhabited beyond just following the water like we have been. And so to do this, a new term has come up called urability. And urability encapsulates a planet's or location's ability to actually host an origin of Earth-like life. And to understand urability, we really need to look at Earth, because that's the only place we know that an abiogenesis has occurred. So a prevailing concept is that mineral surfaces oscillating between wet-dry cycles catalyze the origin of life on Earth. And this is kind of the hypothesis that my work is based off of, as I look at what planetary features might influence such an origin. So if we're thinking about early Earth, for wet-dry cycling, we're really thinking about volcanic islands. And that is because of the phenomena daytime sea breezes and nighttime land breezes that I have pictured here. And what these are is they're breezes that arise due to a temperature difference between land and water due to their difference in heat capacities. So during the day, we have land warming up at a much higher rate than the water next to it, and this leads to airflow and precipitation. And the inverse of this occurs during nighttime when we have the water retaining heat from the day and the land cooling off very rapidly. So even though these breezes are a local phenomena, planetary features influence them through affecting spatiotemporal patterns of surface temperature and precipitation. One such planetary feature of interest is obliquity, or planetary tilt, which I have pictured here. And all obliquity is a planet's tilt on its rotational axis, so in respect to its star. So here I have pictured 0, 45, and 90-degree obliquities. If you imagine a volcanic island at the equator on each planet, you can see that at 0-degree obliquity, it would receive stellar radiation year-round. 45 degrees, it would experience severe seasons, kind of like the seasons we have here on Earth. And 90 degrees, it would be in the middle point between a pole that receives constant solar radiation during its summer and a pole that is absolutely in winter at that time. Also to note on the 90-degree case, that equatorial island would not receive day-night cycling. So that could drastically affect the land and sea breezes that I talked about earlier. So to investigate the effects of obliquity on wet-dry cycling for volcanic islands, I am using a general climate model called Exoplasm. And Exoplasm has a dynamic atmosphere and a slab ocean, and I am using it at a resolution of 64 latitudes and 128 longitudes with 10 atmospheric layers. So here I have some of my preliminary results regarding the mean surface temperature for global scenarios with different obliquities. The black little outlines are little volcanic islands that I have placed and randomly fleshed out with a random walk. So you can see in the 15-degree obliquity, we receive the really cold temperatures at the poles. And then as obliquity increases, we start having an evening out of our temperatures until we get to, say, the 75 and the 90-degree obliquity, where we have extremely warm temperatures at the poles. So this first plot is kind of a summary of that information. It is the mean surface temperature across islands, and I have the latitudes out here. The brightest red is that 90-degree case where we have extremely warm temperatures throughout the year at the poles. And then at the 15-degree obliquity scenario is that brightest blue, and we have those really cold temperatures. This plot is new, as in it shows the mean diurnal temperature ranges across islands. So all that means is that I'm showing here the mean day to night shift in temperature for each island. And you can see it's very interesting that obliquity 15 shows the highest ranges in temperatures. And that is because at obliquity 15, in my case scenarios, you're getting the maximum amount of day having, the maximum amount of land having day-night shifting. Because like I said, with the 90-degree obliquity scenario, during the polar summer, that land is just experiencing solar irradiation. It doesn't get a nighttime cycle. So this is some summary plots of those. And here I have the mean surface island temperature according to obliquity, and you see that increase as we increase obliquity. But this is to summarize that diurnal temperature ranges plot. And it's you can see that spike at 15 degrees obliquity, but you also can notice a dip at 45 degrees. And that is because at 45 degrees, we're getting the most even distribution of stellar irradiation throughout the year. So that means we're not getting as much fluctuations in temperature as we could see in other scenarios. So this is the precipitation version of that lat-long plot I showed you for temperature. So in the obliquity 15, you can kind of see the familiar cycle of having the intertropical convergence zone-esque precipitation band along the equator, and then a little bit of decreasing precipitation, and that's akin to the horse latitudes we have here on Earth. And that's all arising from like the Hadley cell and our normal atmospheric circulation that we're used to. And that structure begins to break down as obliquity increases. And actually, after we reach 45 degrees obliquity, we start transferring heat instead of from the equator to the pole, from the pole to the equator. So you can see that kind of inversing of structure happening in 60, 75, and 90 degrees obliquity. And you can also see that shift in this mean precipitation plot across islands in that the lower latitudes, the lower obliquities have a very spiky structure to simulate that multiple cells in the atmosphere. But as we get to higher obliquity, we have a kind of evening out, and it's not as severe. For the diurnal precipitation ranges, it's very interesting to note that all obliquities have a spike in precipitation ranges at the equator. And that's because at lower latitudes for low obliquity scenarios, we have very intense day-night breezes. So that means that we're having very intense cycles of precipitation and evaporation. And then for high obliquity scenarios, we also have that peak because those low latitude islands are in between a pole that is having a very severe summer and a pole that is having a very severe winter, leading to a lot of convection and increased precipitation. So these are the summary plots for that. You can see the mean precipitation actually and the diurnal precipitation ranges both peak at 45 degrees obliquity. And this is due to the same reason why we saw a dip in the mean temperature ranges at the same point, of that we have the most even distribution of stellar radiation happening. And since we have a warm climate on an ocean, basically all ocean planet, these islands are very small, we have a lot of precipitation being generated year-round because we have the most amount of area being evenly irradiated. And so it's really great to look at how obliquity affects precipitation on a day scale, on a yearly scale, and surface temperature. But it's very important to not just try and apply, connect what we see here with the planetary concepts that I have done so today. If we're interested in seeing what planetary scenarios actually promote a origin of life. It's important instead to apply chemistry. And so what I'm going to be doing is I'm going to be covering a wide range of planetary features with exoplasm, and then I am going to be running a pond model to simulate a warm little pond on each planetary scenario. That is going to keep track of the supposed RNA synthesis that might occur, and we are going to be using that to identify which planetary scenarios might be the most conducive to an origin of life. And that is it.
Okay, we have a couple of minutes, or actually several minutes for questions, so here you go.
Hey, great talk. It's Bryce Troncone, Dartmouth. I was just wondering, so how are you going to take into account basically like your habitable zone of each star type, and then the distance from star of like affecting the, maybe it's tidally locked, or if you have like I guess like ice surface versus liquid surface, and if like you have a substellar pond versus global ice climate.
So yeah, this is actually the full parameter space that I'm going to be exploring. And it does include both G stars and M stars. And for the M star planets, we are not exploring obliquity because we are expecting them to experience tidally locked scenarios. As for assuming that the planets are in the habitable zone, we are assuming that the planets have surface liquid water and we can place volcanic islands on them. So that is going to affect where we think the planets are in relation to their star.
Hi, good talk. Sam Nasruddin, University of Arizona. So yeah, following up on what the previous question, have you looked at tidally locked planets and how that might affect wet-dry cycles? And I guess I have a second question. Are there scenarios in which the cycling is too rapid, for example, and that actually might lead to a reduction in the probability of an origin of a life event?
Yeah, so we're definitely thinking about M stars. It's going to have a drastic effect on wet-dry cycling because you don't have that day-night cycling, so it's going to be a little bit more like the 90-degree obliquity scenario. And we think that that's going to definitely just affect the whole thing. As for too frequent of cycling, we're also interested in seeing if we have like a big storm happen on our island that we're looking at and it's like really severe, we could have all of our evolutionary progress like flushed out. Along with that, like if it's too dry for too long, then we could have too much exposure to UV, or we could lose something to seepage. Like we're trying to like also incorporate the strength and some wind data into that. So yeah.
Hi, Lily Robinthal, University of Arizona. Really cool talk, really neat stuff. I'm curious if you know if there are any sort of parallel studies going on investigating like urility in terms of other origin of life scenarios other than wet-dry cycling, and if y'all are kind of collaborating on other possibilities in terms of origin of life.
As far as I know, no one's using exoplasm or like really looking at GCMs to do that. I do know a lot of different people are very fond of different origin of life scenarios. And so what I've told people is that I am generating a data set of different obliquities, different rotation rates, and all of that can be used for multiple scenarios. The only thing is that if you wanted to investigate like hydrothermal vents, you would have to use a model with a dynamic ocean, like Rocky 3D. So I'm not in contact with people doing that, but I'm very interested in being in contact with people doing that.
Hi, Mrityunjay from University of Arizona. So I was just wondering, like, to what extent, the atmospheric composition of a planet that is either tidally locked or not tidally locked would, alter the results that you see. So,
yeah, that's going to greatly affect basically everything. For this, we're assuming an Earth-like planet, so we're just using like a modern Earth atmosphere or early Earth atmosphere. And it's going to affect everything because the atmosphere is a big place where we get ingredients for life. So we have lightning chemistry, we have chemistry happening when we have meteors coming and impacting. Even when they impact, the plume can create organics for life to occur. So that would greatly affect how we would think that the chemistry is, for example, for the pond model, that would greatly impact how we're tracking the chemistry or how we're even thinking that it would be urable at all.
Any other questions?
So how do you justify like the starting water content of your system? And dang, I have one more. Right? I don't think yeah. I'll come back to you.
Okay.
And like yeah, there you go. Yeah, and stripping of the atmosphere. So yeah.
Yeah. So right now we are just running with this global ocean thing, and that is because we think that it is possible for other planets to experience the same ocean accumulation that we have on Earth, because we're really only thinking about Earth-like planets. There's so many different types of exoplanets out there that could have so many different types of urability that I am very interested in investigating, but currently we're just looking at an Earth-like scenario because Earth is the only example we have of an abiogenesis occurring.
Any other questions? All right, give Haley another round of applause. Okay, and that concludes our last session. And there is lunch, not yet. I mean, we can wait in here if you want.
Lunch will be here soon, but it's not here yet. So people can chill, relax, get coffee.
Yeah. So we've got an hour. For the next session, just make sure you're here like 5 minutes early so we could just start right on time. Also, if you're in the next session giving a talk, please come up and see me. I just want to make sure your slides are all good to go.
All right. While that's loading, I'd like to introduce, we have Justin Park, who's going to tell us about a framework for understanding the origin and fate of Earth's carbon. And you should clap extra hard for Justin because he's one of our organizers and he's done a lot for this event.
All right, can you hear me? Yep. Cool. Well, hello everybody. My name is Justin Park. I'm a PhD candidate at Rensselaer Polytechnic Institute in Troy, New York, about 3 hours east of here, so pretty close. So I am interested in our lab in investigating the volatile evolution of the Earth. So today I'm interested in telling you about a framework for the understanding of the origin and fate of Earth's carbon. So why study carbon? Well, hot take, carbon is important for life. And this is true from the molecular level all the way to cells, and then we can get into complex life such as my dog Lincoln. The use of the term complex is often debated. Now, carbon is also important in driving planetary climate systems. So on Earth, we see evidence of a global carbon silicate cycle. Here we have carbon exchanged to the atmosphere, ocean, crust, and mantle. And this exchange is promoting the climate of the planet through billion-year timescales. We anticipate that this, or a similar type of cycle, could exist on other exoplanets, and we can envision that the climate may be maintained and possibly the habitability of the planet maintained because of carbon. So let's look at the Earth's carbon. Where is it? Well, the carbon that we can observe is mostly trapped in the crust or the mantle. We call this the bulk silicate Earth. There is a lot of debate about how much carbon is trapped in these reservoirs, but in general, there's consensus that it's anywhere from 4 to 6 orders of magnitude larger than the ocean, the atmosphere, and the biosphere. So that's a lot of carbon. Now, we can distinguish between these carbon reservoirs using their carbon isotope ratios. Now, we use the delta notation. This allows us to compare the ratio of carbon 13 to 12 and reference it to a standard. Now, a more negative delta value implies that the carbon isotope ratio is lower or the carbon is lighter. So here on the right, I have the delta value plotted against time for marine carbonates, diamonds, and organic matter. And this is plotted for the last 3.5 billion years. Now, one thing that's really curious is that this carbon and all of these reservoirs show an isotopic signature that hasn't really changed over this time. Thus, the Earth's carbon reservoirs have likely been set isotopically for most of the Earth's history. Importantly, what this tells us is that the bulk silicate Earth has a carbon isotope ratio around -5 per mil. And it's been that way for at least 3.5 billion years, probably more. How does Earth's carbon compare to carbon in the solar system? Well, the bulk silicate Earth has a roughly 0.01 to 0.02 weight percent carbon. That's the amount of carbon per kilogram of material. This is actually a lot lower than other things we see in the solar system, such as the sun, chondrites, or comets. Now, another interesting observation, this carbon seems to be isotopically heavier than most things we find in the solar system. The Earth, the bulk silicate Earth, has a delta value of -5 per mil, whereas everything else, except maybe comets, has a lower and lighter carbon isotope ratio. So if Earth's carbon is heavier and it contains less carbon per kilogram than the material that it might have formed from, then that carbon must have left the Earth, and that light carbon, or it is either hidden in the Earth somewhere. Ooh, the formatting got a little screwed up. So one thing I didn't reference is the Earth's core. We think that the Earth's core could have a substantial amount of carbon for a couple of reasons. Now, the first observation previous studies have suggested is that carbon is a highly siderophile element. It likes to be in metal melts. So because of this, there's a hypothesized up to 10 to the 23rd kilograms of carbon trapped in the core at relevant temperature and pressure conditions. A second observation is that from seismic data, we can see the core is roughly 10% less dense than what we expected it to be based on an iron and nickel mixture. So this suggests there may be other light elements trapped in the core. We could have hydrogen, oxygen, sulfur, maybe nitrogen, or carbon. So because of this density deficit, we're looking at specific phases to see what might cause this. Iron carbides seem to have a good density to investigate this specifically. With these estimates, they predict anywhere from 0.5 to 4 times 10 to the 22nd kilograms. So to put this into reference, this is anywhere from 90 to 99% of the Earth's carbon trapped in the core based on these estimates. That's a lot of carbon. So we can't sample the Earth's core. It's a little too far down. But we can sample cores of other differentiated bodies in the solar system. Here we can look at iron meteorites. Now, iron meteorites, despite being iron, have a lot of carbon in them. And this carbon comes in different phases. There's carbon in graphite nodules like this. There's also carbon trapped in the metal phases themselves, such as taenite and cohenite down here. Now, this carbon would have been sourced from the overlying silica from the planetary embryo. So we can look at that carbon and see, is it fractionated between these phases? The carbon in the graphite likely contains the same isotope ratio from the silica. They precipitate in equilibrium. This has an isotope ratio around -8 to -5 per mil. Now, the carbon in the metal is slightly different. This is an isotope ratio of -22 to -18 per mil. So there is a fractionation. What this is telling us is that the lighter carbon is preferentially stored in the metal phase. This has really important implications for the Earth's core. So we can compute a fractionation factor. This is effectively the difference in delta values between these phases. For this iron meteorite, it's around -13. Now, this fractionation factor does have some theoretical backing. Here, what I am showing is the fractionation factor plotted against temperature on the x-axis. And this is for a variety of different phases and expected carbon compounds in the mantle equilibrating with iron carbide. Now, one thing to note here is there is a temperature dependence. As the temperature gets higher and higher, there is less fractionation that's expected. Importantly, though, every single one of these fractionations is below 0. So regardless of the temperature, regardless of the phase, this tells us the iron phase will have a lot more light carbon. So we can use this to kind of study maybe the Earth's core formation. We know iron meteorites probably differentiated at a lower temperature. Thus, their fractionation is a lot higher, or greater magnitude. But we also know that the Earth's core likely formed around 2200 to 3200 degrees Celsius. That means we should get significantly less fractionation. And we expect between -1 and -4 per mil fractionation for the Earth. Now, if we want to model this, we need to look at the Earth in the early stages of its evolution, specifically the magma ocean. In the magma ocean, there would have been metal droplets that are swirling around and equilibrating with the silicate and kind of consuming and absorbing up carbon. These metal droplets would eventually accumulate at the silica solidus, and as metal is denser than silicate, they would release a instability and travel into the core through these large diapirs. So incrementally, the core is built, but also incrementally, we're adding carbon to the core through these diapirs. We can model this a little bit further. At time TD, that's when differentiation begins. We start to see carbon is released from the mantle and brought to the core. TD finish is when we see differentiation ends, and we have the set conditions we observe today. So the mantle carbon decreases, core carbon increases, but the total carbon on Earth doesn't change at all. We can look at the isotope ratios and see something as well. As we begin here, the isotopically light carbon will be transported into the core. Due to mass balance, this will start to make the mantle heavier and heavier, and as that material is transitioned back into the core, we're going to start to get the core heavier and heavier again. So this blue line here is what we observe in the bulk silicate Earth, a value of -5 per mil. This modeling implies that the carbon that the Earth formed from and the carbon in the core are isotopically lighter than what we observe today. Modeling this a little bit more thoroughly, we can use a Rayleigh fractionation to explain these results. Now, on the y-axis here, I have the percent of Earth's carbon in the core versus the isotope ratio of potential materials that gave Earth its carbon. One thing to note, there is an absolute limit here. We cannot go above -5 per mil. That's because our fractionation is always negative. Now, because of this, we can also look and see how many steps it might take to deliver that carbon to the core. With ultralight materials, say solar carbon, it takes a lot more steps to deliver core carbon, and we get a lot more carbon in the core, upwards of 95 to 99%. So in total, Earth's starting isotope ratio has to be less than the bulk silicate Earth. Here we can also model the temperature dependence of this. Here I've plotted the percent of carbon in the core versus the fractionation factor, and this is temperature dependent. Remember, the fractionation is reduced as we increase in temperature. As both of those happen, we get more carbon into the core as more steps are necessary to reach the observed isotope ratios we have today. So we can model this in three-dimensional space, as there is potentially infinite combinations of fractionation factors and starting materials. That's what I've done here. Again, the x-axis is the starting isotope ratio, the y is the fractionation factor, and z is the percent carbon in the core. The red lines here represent isobars of equal carbon concentration in the core. And I've plotted for reference some of the starting materials, such as solar carbon or chondritic carbon. We only care about a small window of this for the Earth, so we can look in this range. This is appropriate temperature pressure conditions. And then we can add relevant reference materials that the Earth might have started with. Now, on this right plot here, I have, again, the Y, the carbon content in the core. Left axis is in kilograms, right axis in percent. And the x-axis is the delta 13C of the starting material. Each star represents an individually distinguished starting material, such as chondritic material, solar carbon, or a mix of both. We can start to use this to differentiate which material might be useful and form the early Earth. We know that the Earth has a mass, and it's below what we see the carbon content of the solar experiment should be. So we can rule this out and say it's not going to happen. We can also look at the other experimental techniques of investigating carbon in the core, and we see that the chondritic material is plotting right within this range. So finding one, we suggest that based on isotope ratios, the core should contain the majority of the Earth's carbon. We also suggest that this is likely a mechanism for identifying chondritic material as the formation of Earth's carbon. We can kind of flip this on its end and investigate the bulk carbon contents of the iron meteorite and their parent bodies. We can look at the fractionation factor we have plotted up here, and we can predict that the carbon trapped in these bulk embryos might be roughly 2/3 in the core and the remainder in the rest of the silica. So we can do a kind of meteorite forensics. So there is a fun thing that can happen and complicate this. Any carbon delivery after core formation will require additional mass balance. We have evidence that there are siderophile elements in the mantle that aren't in the core, and this tells us that there have been some delivery elements after this. A possible explanation for this is through a late veneer that could have delivered carbon from 0 to 50% of what we observe in the bulk silicate Earth. So adding to our model here, if carbon is added following differentiation, it'll only be in the mantle shown here. It won't increase the carbon in the core, and then it will increase the total Earth's carbon reservoir. We can model this isotopically, and the late veneer likely was chondritic, so it's supplying lighter carbon sources than the bulk silicate Earth. And kind of counterintuitively, this implies more carbon would be in the core, as there is a mass balance now where this step here is a heavier carbon than what we observe today. So more steps are necessary to get that carbon in the core. Now, what you're about to see is a model where we incorporate that to kind of characterize all of the possible amounts of carbon delivered through a late veneer. We animated this. Adding on the right now, we have a plot of core content versus the fraction of the bulk silicate Earth carbon delivered following differentiation. So in this model, or in this animation, you can see that the carbon contents are increasing as we add more carbon following the late veneer. Now, this is telling us that the starting materials might be good if there is no late veneer, but if we have a late veneer to a certain degree, they will eventually promote too much carbon in the core, or imply too much carbon in the core, to the point where they are no longer acceptable starting materials. I'm going to play this one more time. This time, let's focus on this boundary here. When we look at this, we can see it's expanding towards the right. That means the earlier materials forming the Earth could have been heavier with their carbon isotope ratio. This implies that we could have a mix of chondritic or cometary matter forming the Earth's carbon reservoirs. So with this, a chondritic late veneer implies that more carbon can be trapped in the Earth's core. We have shown this through the animation. We also know, based on reasonable bounds of our late veneer, that these are still consistent with what we're observing in solubility and density estimates. That's this region right here. A third finding is that the late veneer allows for the earliest sources of carbon to the Earth to be heavier than the bulk silicate Earth that we have now. So this is higher than the 5 per mil value. That's illustrated by this blue curve here that could be this 5 per mil. So with this, we can get a preferred solution. With a roughly chondritic starting material, a delta value of -15 plus or minus 2, and roughly 15 to 25% of the Earth's bulk silicate carbon delivered in this late veneer, we predict that the carbon content in the core would be anywhere from 1 to 2 times 10 to the 22nd kilograms. That's, again, a lot of carbon, but to put that in reference, it's 95 to 99% of the Earth's carbon trapped in the core. So in summary, the isotope mass balance framework that we've developed here, it's allowing us to extrapolate what the sources of Earth's carbon could have been and how much of that carbon could have ended up in the core. We find that the most likely source of this is chondritic material, and then the carbon in the core could account for 95 to 99% of the planet's carbon. We've shown that this can be applied to other differentiated bodies in the solar system. We can do a sort of meteorite forensics investigating the bulk carbon content throughout the solar system. And finally, this proves that most of the Earth's carbon is not available to life. This is pretty powerful, as this carbon wouldn't be there for the origin of life, the evolution of life, or any associated chemistries. It's important to think about this in a planetary context, because this implies that life found a way, even though it seems the odds are stacked against it. With that, I'd like to thank you all for listening. I'd like to give a shout out to everyone back home at RPI, and a quick thank you to our funding, the Earth First Origins project from NASA. Thank you very much.
Hi, great talk. Do you suppose that the late Veneer mechanism that brought a lot of carbon to Earth is sort of normal for our solar system? And can you speculate, perhaps, if there's a time component to the availability of carbon to sort of help with the abiogenesis?
Yeah. So the idea of the late Veneer, we find evidence for it both on Earth, the Moon, and on Mars. The general idea is having some form of differentiation occurring first, and you still have these impactors hitting the Earth, Mars, or the Moon. This will add material to your planet far after the time period of when you should have carbon abundant in your silicate mantle. Sorry, what was the second question as well?
Oh, yes, the contribution of this late Veneer carbon to abiogenesis.
Gotcha. So if we look at the carbon reservoir on the Earth, a lot of what we see could have been delivered following core formation. Depending on that X variable, how much carbon is present, that would be carbon that could all be usable by life organisms. This could imply that the majority of Earth's carbon was delivered in a late Veneer. If that's the case, then that might say that carbon is available to life. There's not really a way to distinguish now using just these isotope ratios how early this process happened. This is just summarizing how much carbon content has been delivered to the Earth following core differentiation. So for the origin of life studies and abiogenesis, it's still a little unclear based on isotope ratios, but at least certainly before 3.5 billion years ago, there would have been a carbon reservoir that had the appropriate contents.
Hey, Justin. So, really cool talk, by the way. Would this be an Earth-specific thing, or would you expect this on any rocky planet? And if so, how would that impact the search for potential biosignatures on other rocky planets?
I definitely don't think this is an Earth-specific thing. However, it could be a size-dependent thing. Mars is a lot smaller than the Earth, and it's expected to have formed a lot quicker. So the actual magnitude of carbon that's trapped in the Martian core could be very different than what we observe on the Earth core. Further with that, the timescale Mars took to form is significantly shorter than the Earth. So the chondritic material that might have been delivered could represent a much larger portion of the planet's carbon. Speaking specifically to the evolution of life anywhere, it seems if you have a lot more carbon being delivered later, you could have, say, for Mars, that carbon would be more readily available to the organisms that could thrive there. So if you have a kind of mass dependence here, the smaller planets might have a lot more carbon available to them from chondritic material. Larger planets might have more carbon or less carbon available following core differentiation. Does that answer your question?
We have time for one more.
Hi, Satish from University of Naples. I just want to check with regards to the iron meteorite references and like the chondritic ones that you've used in the visualization. Oh, by the way, cool visualizations. Are they the same or?
Sorry.
Because like here you used the iron meteorites as a proxy.
Mm.
And then later, when it comes to the models and stuff, looking at chondritic ones, are you looking at iron stony meteorites? Just to clarify, are they different?
No, so the carbon sources that we use in the model are generalized. The isotope ratios that are trapped in these meteorites, they range anywhere from -5 to -30. So I was trying to put up a range of potential chondritic materials. These iron meteorites don't really deliver a lot of carbon, we think, to the planet. We think it's probably the more basic level meteorites that aren't differentiated, so our carbonaceous chondrites, that kind of stuff, delivering specific carbon nutrients to the planet. Cool.
All right, and next we'll welcome Sam Pryor, also from RPI, and she'll be giving a talk on enhanced phosphodiester, my new word of the day, bond formation in abiotic RNA polymerization via a cycled microdroplet synthesis setup. It's a tongue twister.
You're telling me? Not yet. Whoop, that is the end of my talk. If you just click 1 and then enter, it should take you to the first slide. Close your eyes, no spoilers. Okay, cool, this is the beginning slide. Hi everybody, can you all hear me all right? Cool. All right, I'm Sam Pryor, third-year grad student in the Department of Earth and Environmental Science at RPI. Working with Dr. Jake Shelley and the Rare Center, focusing on origins of life and prebiotic chemistry. Specifically, I'm interested in microdroplets as a sort of unique reaction environment, focusing on how they can impact RNA oligomerization, or phosphodiester bond formation. But before we get too much into what I'm doing, I wanted to provide a quick primer for prebiotic chemistry. Most of you probably already know about prebiotic chemistry at this point, but I thought better safe than sorry. We'll have a quick review here. So I like to think of prebiotic chemistry as a sort of chemical evolution scale, with molecular complexity going from least complex to most complex here. So we would have started with the inorganics present from Earth's formation, like all that carbon that Justin just told us about. These inorganics would have undergone geochemical processes to eventually form simple organics, and then increasingly complex organics until we get to the building blocks of life that we all know and love, like amino acids, sugars, and our nucleobases, sides, and tides. These building blocks would then undergo condensation reactions and form some sort of biopolymers. So, we have our lipids that we've heard all about today, nucleic acids that we've heard about, and proteins that we've heard all about. These guys would then somehow form the earliest life. Now, I know we're probably not all chemistry minded here, but there's a really, really wide range of different chemistries here, and I'm just one grad student, so I'm only going to focus on one little chunk of this. The transition from building blocks to polymers, which, as I mentioned, is mostly facilitated via condensation reactions. But to get some more context for our prebiotic chemistry, we need to first think about chemistry in the context of environments, specifically Earth's environment, since we're interested in the origin of life. So we have the proto-Earth over here that was likely hit by a large impactor called Theia. Crash, boom, bang. We get a bunch of stuff happening, and then eventually we settle into the Earth, which has all the material that makes up the modern Earth, and the Moon. Now, it's important to note that this Hadean Earth would be very different from the Earth as we know it. I mean, environmental conditions are going to be very different, but maybe not as different as we think. So if we think about the Hadean Earth in terms of geologic timescales, since we're interested in the origins of life, we're interested in this sort of earliest chunk of Earth's history over here, the Hadean, the Archean, really early on. About 100 million years after the Earth's formation, or the moon-forming impact, rather, recent evidence suggests that shallow water oceans could have formed. So Justin was telling us all about the magma ocean and differentiation. So relatively quickly on the geologic scale, we could have had global water oceans pretty quickly. And with the formation of water oceans, we get microdroplets, which are micron diameter sized water droplets that are going to be abundant anywhere where there's water on the Earth's surface. This is because they form by wind and wave action. So in this figure down here, we see microdroplets being formed from a crashing wave, say, in the ocean. Conversely, we also get bubbles that get trapped under the surface of the water here that are going to provide even more environments for the interesting chemistry that we can also see in microdroplets. So we get microdroplets, we get microbubbles. But importantly, this stuff doesn't just happen over the ocean. It can also happen over lakes. It can happen as a result of hot springs, geysers, or any sort of, violent things, maybe like impactors crashing into the ocean or a lake can kick up microdroplets, or even like clouds. We all know and love clouds. So you can get condensation on nuclei in the atmosphere that can also give microdroplets. So they're going to be really, really, really abundant anytime you have water on the Earth's surface. In addition to being really abundant, they provide some really unique chemical properties, mostly due to the partial solvation state available to reactants at the interface. So if we were to plot a reaction progress diagram for this reactant partial solvation state, we see reaction rates that are more akin to gas phase reaction rates than they are to liquid phase reaction rates. However, our density of our reactants is higher than the gas phase. It's more like the liquid phase. So that partial solvation state is going to kind of give us like the best of both worlds between gas phase and liquid phase chemistry. So I mentioned before, I'm just one grad student. I'm focusing on one very small section of prebiotic chemistry, specifically condensation reactions. I decided to look at RNA formation because there's a wealth of literature on it, mostly because of the RNA world hypothesis that we've kind of heard mentioned today. But condensation reactions, including RNA formation, are going to be a really important step to increase molecular complexity. However, there are some issues with them occurring in bulk solution, mostly because they result in the production of a water molecule. So if you have bulk solution and you have a reaction trying to proceed that's going to produce a lot of water, it's not going to really want to go. The reverse reaction, hydrolysis, is actually going to be preferred here. So I should have said, with nucleotide formation, we get phosphodiester bond formation. But hydrolysis is going to be preferred in bulk. So we need to come up with some way to sort of drive this reaction, whether it's in bulk or wet-dry cycling or some other method. Now, like I mentioned, this reaction has been studied since like the 1960s for prebiotic chemistry. So a lot of really smart organic chemists got together and came up with a lot of ways to drive this reaction in bulk. My personal favorite is using pre-activated nucleotides. So if you take an imidazole group, which we have over here, and just kind of slap it onto the phosphate there, you're going to get your reaction to proceed because imidazole is a really good leaving group. So as soon as you get your pre-activated nucleotide in solution, that imidazole, it's ready to go. And as the imidazole leaves, you can get the energy that you need for phosphodiester bond formation. So with our pre-activated nucleotides, we're going to get oligonucleotide formation in bulk solution. Scientists came up with this around like the 1960s, 1970s, so this reaction's been around for a while. So there's a lot of literature on it, which makes it a really good reaction to study. I should mention adenine is typically the nucleobase used because it is the easiest to oligomerize. Some of the other nucleobases have some unique experimental difficulties with oligomerization. But this reaction, since it's been studied for so long, makes it a sort of like a model condensation reaction to look at in microdroplets. So we're going to take our reaction, we're going to put it in microdroplets, and we're going to see, can microdroplet chemistry impact RNA formation? But really what we're getting at here is where is the reaction actually occurring in the droplet? And we sort of have two options here. One, that the reaction is occurring at the surface of the droplet, where we can get that partial solvation state available to the reactants, so we can drive this really unique chemistry. And if the reaction is occurring at the droplet interface, we would expect some sort of enhanced RNA oligomer formation or some sort of enhancement in our oligonucleotide formation. However, the reaction could also be occurring towards the interior of the droplet, where it's mostly just bulk, so you wouldn't really expect any difference between your bulk controls and your microdroplet reactions. So I got at this in the lab. I'm an experimentalist, so I designed some experiments to test my hypotheses here. I used two separate independent nebulizers here to make my microdroplets. Over here on the left, we have an ultrasonic nebulizer. And over here on the right, we have a vibrating orifice aerosol generator, or VOAG. It's really important to use two independent nebulization techniques because they're going to make droplets in different ways. So you can compare your results between the two nebulizers and see if it's a nebulization-dependent effects or if it's actually a result of the microdroplet chemistry, which is the heart of the question that we're trying to get at here. Importantly, Both nebulizers produce droplets that are a similar size range, so that shouldn't impact the chemistry too much. I will note the ultrasonic nebulizer is the preferred nebulizer because it is much easier to use. This one kind of heats up and shuts itself off every 25 minutes, which is super fun to deal with in the lab. Also, this one is more robust. I can run it 24/7 for potentially weeks on end. I'm going to walk you through how I set up an experiment. I take both nebulizers. I have my solution phase with my pre-activated nucleotides, sometimes with the salt, depending on what sort of environments I'm trying to mimic. Turn on the nebulizer and make droplets, and then I collect them either in a spray chamber over here with the ultrasonic nebulizer, or my super duper high-tech product reservoir here that is a 50 milliliter Falcon tube slapped on the end. Then I collect the condensate and cycle it back into the solution reservoir to get this sort of natural cycling between the bulk phase and the microdroplet phase, which is going to mimic what we see in the cycling that we see in the ocean, but also geysers, hot springs, lakes, and clouds. So really the overall goal is trying to see how our chemistry, see if we can better mimic natural environments in the lab to really get at the heart of our research question. To analyze my samples, I use matrix-assisted laser desorption / ionization time-of-flight mass spectrometry, or MALDI-TOF-MS, or just MALDI. And this provides data that looks like this. So we have signal on the y-axis as a function of mass-to-charge ratio on the x-axis, sort of just a standard mass spec. Importantly, all of these peaks that you see here are oligomer peaks. So we have everywhere from a dimer up to a 7mer. And I mostly look at this highest peak, so the maximum detected oligomer length for each spectrum. So I collect those for all of my experiments at different time points throughout the experiment to get some sort of like quasi-kinetic data. And then I average all of that, and I get a plot that looks sort of like this. So we have average maximum oligomer length on the y-axis versus a function of time on the x-axis. So here I'm looking at just pure water with our pre-activated nucleotides in solution. And we get some really interesting results. We see that microdroplets are impacting RNA oligomerization. If we look at the shortest time points, we see that the longest oligomers that are formed in microdroplets form very quickly. So within an hour, we make the longest oligomers that we see in droplets in just pure water with no additives. If we look at our later time points, we see that so after 3 days, microdroplet-produced oligomers are about 3 times longer than those seen in bulk. These results are sort of unprecedented for just pure reactions with no additional catalysts or intermediates or anything. Typically, in bulk solution, you'll get maybe dimers or trimers, but we see that with microdroplet formation alone, we get a potentially 3 times increase in oligomer length. However, when we add salts to sort of mimic that ocean environment, our data gets a little bit more nuanced. So you can see at later time points, the bulk is actually going to produce longer oligomers. It takes longer for our bulk reaction to get to equilibrium. So, oligomers in bulk with calcium chloride, which is the salt I'm using, can regularly be like 10 nucleotides in length at later time points. But we don't see oligomers that long in microdroplets. However, if we look at earlier time points, we're seeing that microdroplets are actually producing longer oligomers faster than bulk solution. So this is implying some sort of kinetic rate enhancement of microdroplet formation. So if we zoom into that shorter timescales, we can see, once again, the longest oligomers that occur in microdroplets occur very quickly. So here within 2 hours in the presence of salts. So even with salts, microdroplet formation is doing something. It's still enhancing RNA oligomerization. It's just at shorter timescales, and we're not seeing oligomers as long at later timescales. When we compare data from both of our nebulizers to sort of verify that the trends that we're seeing are as a result of microdroplets and not the specific nebulization technique, we see similar trends. So once again, the longest oligomers that are occurring in microdroplets occur very quickly, for 2 hours, within 2 hours for the ultrasonic nebulizer, or within 4 hours for the VOAG. Whereas if we look at the bulk solution phase, we see comparable length oligomers are only formed after 6 hours or longer, which indicates that microdroplets, once again, are doing something independent of nebulizer type, even in the presence of salts. So in summary, microdroplets are really common, ubiquitous reaction environments. They're prebiotically plausible. They're going to be around as long as you have water on a planet surface. It doesn't even have to be Earth. And they're really unique reaction spaces that haven't been explored very much for prebiotic chemistry. Here I showed that microdroplets do in fact enhance RNA oligomerization with oligomers forming very rapidly in droplets. Importantly, we found that even in just pure water microdroplets with no additional additives, we get up to 3 times length enhancement compared to bulk controls for our oligomers. And all of these data suggest that the reaction is likely occurring at the droplet surface, with the partial solvation state playing a role in the reaction kinetics. Future work hopes to explore alternate nucleobases in addition to adenine. We want to look at varied salt concentration and composition to see if that impacts RNA oligomerization. And then we also want to investigate alternate leaving groups, so in addition to imidazole, to see how that might impact how or where the reaction occurs in droplets. So with that, I'd like to thank everyone back home at RPI. Thank all of you, thank the organizers, and I'll take questions.
Hello. Maybe I missed that. Could you tell me what the concentration you're using for this oligomerization thing, and what buffer you're using? And the second question is that for your system, for like when you're using nebulizer and like recycle your solution, what's like the container is? Is that like made of glass or something?
Sure. So to answer your first question, I'm using 15 millimolar for my preactivated nucleotides and 1 molar salt concentration. What was the second part of your first question?
What's a buffer you're using?
I'm not using a buffer. So I'm trying to make it as simple of a system as possible just to see if microdroplets have any impact.
The second is that when you're cycling stuff, what was
So for the ultrasonic nebulizer, it's a glass spray chamber. For the other one, it's some sort of plastic. I'm not sure the specific plastic composition.
like what I'm seeing for my experiments, because I'm using radiation, so I can tell if I'm using glassware, it will stick on the surface.
Okay.
So we'll do some. Is that possible? Your stuff is sticking on the surface, or like do you use like stuff to prevent that from happening?
No, it definitely could be possible. Since I'm looking at the condensate, the way the spray chamber is designed, it's meant to sort of collect all of the condensate and it'll funnel down. So it's like the reaction is always, or the solution is always cycling through. So I wouldn't expect anything to get stuck to the spray chamber and stay there. Yeah, I wouldn't expect it to, but that would be an interesting thing to kind of check out.
Okay.
Thank you.
Thank you.
Thank you for that.
Yeah. Hi, Alex Deansrow, Georgia Tech. I'm curious how generalizable you think the kinetic effects of the partial solvation interface might be to like other potentially prebiotically relevant reactions like oligopeptide synthesis or depsipeptide synthesis?
Great question. So it is very reaction dependent. So you can't really generalize the specific reaction rate enhancement that I'm seeing. I know there has been previous work by some other people looking at peptide formation in droplets specifically, but for other sort of reaction classes, it's still largely unexplored.
Thank you.
Yeah, thank you.
Hi, super cool talk. This was really great. Hi, Pesha Medavia from ASU. I had a really similar question, but on the other axis. I was curious if you think similar effects might be observed if the solvent is different. So I was thinking about like the tholins on Titan, how you have these little aerosol droplets, but it's not water, right? So yeah, I was just curious if you had any thoughts about that.
Absolutely, it would play a role. So really, what's going to drive a lot of the surface versus bulk chemistry in the droplet is going to be hydrophobicity of your reactants. So if you're changing your solvent, the hydrophobicity of wouldn't be hydrophobicity anymore, right? It's going to depend on the chemical properties of your species of interest interacting with your solvent. So that's a great question, and yes, it will impact.
Cool. Okay, thank you so much.
Yeah, no problem.
Hey, Sam. Good talk. Super interesting. Abby Deering, Georgia Tech. I was wondering, you talked about the reaction happening at the droplet surface versus the interior. did you, maybe I missed it, but how are you determining whether or not it's happening at the surface or the interface?
Yeah.
Or no, the interface or the interior?
So I'm running bulk, I didn't mention, I am running bulk controls with all of these. So basically, it really comes down to this. If I see an enhancement in the solution that underwent microdroplet formation, then I'm assuming that it's as a result of something happening at the droplet surface. Because if the reaction was just happening all in the bulk phase, you're not going to get any of those reaction enhancements. You would expect very similar. So in the data over here, hold on, we zoom in. In the data over here, you would expect instead of being up here, the oligomer lengths would be down here, much more similar to what we would see in the bulk controls.
That makes a lot of sense. Thank you.
Thank you.
Hi, a really cool talk. Juliana DiGiacomo, Georgia Tech. So my question was kind of about, in your experiments, you were seeing reactions on the timescales of hours into like up to days. And just, this is more on the feasibility of droplets on the prebiotic Earth. Like, what did you think, do those longer timescales kind of become less feasible in terms of a lifetime for like a droplet to be before it like re-enters into bulk solution?
That's a great question. The droplets are going to have a residence time in the atmosphere that's largely dependent on their size. So for small droplets like what I'm looking at, in the tens of microns or less, it's going to depend on air currents and stuff like that, but it's not out of reason for them to be in the atmosphere for days to weeks, maybe even years if they're really small. You can also get some really interesting things that happen, like your droplets can go up, all the solvent can evaporate, and you can be left with a particle that can then get rehydrated if it goes into a more humid sort of environment. So longer timescales could be feasible, but shorter timescales are also feasible for droplets, which is part of the beauty of them.
Thank you.
Just a quick question, I might have missed it. So in like this plot here in the bulk, is that just in like a test tube or a beaker?
Yep, just in a microcentrifuge tube on the bench top.
So why could why couldn't the reaction take place inside the microdroplets if the concentration is like increased greatly?
Sorry, could you repeat that?
Why can't the reaction happen inside the microdroplets versus on?
Oh, sure. So if the reaction was happening at the interior of the droplets, you're not going to get that partial solvation state playing a role. You would expect results that are more like the bulk phase. So it really comes down to like how much water is hanging around. So at the surface, there's less water. So you're going to get differences in your reaction. We can talk about it more later if you want. I can explain it better later.
Okay.
Cool. I think I'm probably running out of time. Yeah. Thanks.
All right, and welcome Rohan. He's going to tell us about photosynthetic biosignatures, biogenetic oxygen production under far red light, and transcriptomic response in cyanobacteria.
Oh, there it is. Okay. Cool. Cool. Yeah, sure.
Next up, we have Michael Wong with a novel in situ biosignature technique based on Pyro, GCMS, and machine learning.
All right, thanks. Yeah, it's so great to be here at my first AbGradCon. Really excited to tell you a little bit about the work that I've been doing for the past couple of years at the Carnegie Institution of Science, where I'm a NASA Sagan Fellow. So as we all know, planetary exploration is embarking on a new golden age of astrobiology, where numerous spacecraft missions will be tasked explicitly with trying to find evidence for habitability and life in our solar system. We're in this saga of a Mars sample return mission. Europa Clipper launches later this year. Dragonfly will visit Titan in the 2030s. And as we heard about from Morgan Cable, we're thinking about future missions to Enceladus to try to understand whether or not there's life on that icy ocean world around Saturn. So I think now is really the time to try to develop some really cutting-edge biosignature detection techniques for solar system exploration. For me, any strong biosignature detection technique has four main attributes. First, it has to be robust, which means immune to false positives and false negatives. Second, agnostic, which means it's capable of identifying life as we know it and also life as we don't know it. Probabilistic, which means that we can quantify our confidence in having detected or not detected alien life. And finally, flight ready, which means it utilizes instrumentation that NASA, ESA, or some other space agency will actually strap onto the back of a rocket and launch into space in the near future. So the biosignature technique that me and my colleagues have been developing at Carnegie seeks to satisfy all four of those central criteria. This is a robust, agnostic biosignature technique based on machine learning and pyrolysis GCMS. We published the initial paper on this technique last September in PNAS in a paper that was lead authored by my friend and colleague, Dr. Jim Cleaves. So imagine for a second that you want to find life on Europa. What are you going to do? You're probably going to send a spacecraft to sample and characterize some of that surface material on this world, this plume detritus that most likely originated in the habitable subsurface ocean below. You might launch an instrument called a pyrolysis gas chromatograph mass spectrometer. Basically, what this is a very fancy oven that not only bakes your sample, but tastes it for you too. And these types of instruments are sent to all kinds of interplanetary destinations. As we speak right now, there's one sitting on the Curiosity rover on the surface of Mars. So the data that a pyrolysis GCMS gather look like this. On the x-axis, we have the mass-to-charge ratio of various ions that are being counted by the detector as the sample heats up with temperature through time. So the z-axis on this plot is just the intensity, the number of those molecules being counted per second at that particular mass-to-charge ratio at that particular temperature in the chamber. Now, if you found something that looked like this on Europa, I think you'd be very excited because this is what the average Earth bacterium looks like. Thing is, about looking for life on an alien world is that there's no guarantee that it will use the exact same biochemistry as life on Earth. So it might be naive to design your life detection experiment around, trying to find that particular peak or this particular molecule over here. Instead, there might be something about the overall distribution of organic molecules in this sample that helps us determine whether or not it was alive or never alive. And so what we've been doing at Carnegie is pyrolyzing and running through our GCMS all kinds of organic-rich samples, things that were very recently alive, like bacteria and plant matter, things that were once upon alive, but over millions of years have been geologically processed into petroleum, oil, coal, shale, etc., and organic-rich samples that were never alive, like carbonaceous chondrites and the results of Miller-Urey type origin of life experiments. Now, I hope that you can appreciate just by eye, looking at these various distributions of organic molecules, that the distribution of organic molecules in all of these samples are very different from one another. And so when we saw this, we wondered, hey, can we quantify how similar or different these distributions are from one another, and also train a machine learning algorithm to tell the difference between a sample with biogenic origins and a sample that was completely abiotic. And so that's exactly what we did. We trained what's called a random forest algorithm on 134 diverse organic-rich samples. And the final algorithm is able to tell the biogenicity of a sample that it's never seen before to an accuracy of about 90%, which is absolutely astounding. Not only can it tell something that was alive versus not alive, it can also distinguish between degraded and fresh biotic matter. And so what you're looking at here in this plot are the first 2 of 20 principal components of our data. You see the abiotic samples all cluster in this tight region over here, with the biotic degraded samples sweeping up this way and the biotic fresh samples sweeping down here. Now, you might be a little bit worried about the overlap in this region, but remember, these are just the first 2 of 20 principal components. So if you imagine extending this graph into a 3rd or 4th or 5th dimension, these clouds of points will further segregate, hence the 90% accuracy of our algorithm. Now, what's really exciting for me as an astrobiologist is the potential to use this technique to look for what we call biological anomalies on other worlds. So imagine you go to Europa, Enceladus, Mars, Titan, etc., and you find organic-rich samples that plot somewhere here or maybe out here. They have organic distributions that are very different from all known abiotic samples, and the organic distributions are also markedly different from the organic distributions in life as we know it, whether fresh or degraded. I think if we saw something like this, we might be looking at a potential biological anomaly of life using a very different biochemistry from life as we know it here on Earth. So with this technique, there are 2 big frontiers ahead of us. The first, obviously, in astrobiology, we now have this robust molecular technique of looking for life, including the identification, potentially, of life as we do not know it. We also have this amazing opportunity in paleobiology. For the past several decades, paleobiologists have been at each other's throats about these ancient 3 to 4 billion year old ancient Archean cherts or other ancient rocks that some groups say have traces of microfossils and other groups say no way. And so maybe our machine learning technique will be able to shed some light using an independent line of evidence to try to understand the evolution of early life on our very own planet. Okay, so why fundamentally should the distribution of organic molecules in living samples be different from the distribution of organic molecules in non-living samples? To answer this, I want to make an analogy to statistical mechanics. So imagine you have a hot cup of tea. The microstate of that cup of tea will be given by the position and the momenta of all the particles in that teacup. And the number of microstates that correspond to a cold cup of tea in a room temperature room are vastly greater than the number of microstates that correspond to a hot cup of tea in a room temperature room. And this is why gradually over time, your hot teacup will cool down. This is our second law of thermodynamics. This is our arrow of time. So the analogy that I want to make to life is this. Maybe there is a high-dimensional phase space not of position and momenta of particles, but of molecular patterns that helps us differentiate between living samples and non-living samples. Now, the curious thing about life is that it seems to evolve in the opposite direction of what the second law of thermodynamics should say. We have origins of life. Non-living worlds transform into living worlds with global biospheres, increasing seemingly the complexity of that world with time. This seems very puzzling until you remember two crucial facts. Number one is that life is an open system, able to exchange matter and energy and information with its environment, and also that life responds to selection pressures, in particular selection for function. So we have a companion paper out also last year, published in PNAS, that is titled On the Roles of Function and Selection in Evolving Systems. It tries to get at the more theoretical nature of what might be going on here. This is a paper that I'm absolutely proud of because it represents more than 2 years of very intense discussions, not just amongst an interdisciplinary group of scientists, but also between scientists and philosophers of science. So Carol Cleland, for instance, and Heather Demarest, these are professors of philosophy of science at the University of Colorado Boulder, who really helped sharpen the ideas in this manuscript. So the purpose of this paper was to try to introduce an explanatory framework for the evolution of physical systems writ large, so including but not limited to biology. The second law of thermodynamics is the only time asymmetric law so far articulated in our scientific canon. But the second law does not explicitly address the tendency for evolving systems to become increasingly complex with time. And so, therefore, a missing law of cosmic complexification, if one does exist, must be consistent with the second law of thermodynamics, but may not inevitably follow from it. So we began our investigation by noting that all natural laws are built around conceptual equivalencies. For instance, Newton's law of gravity was built upon this property of having mass. This property is shared amongst a diverse group of phenomena, whether it's an apple falling from a tree, a planet orbiting a star. Even atoms and molecules are governed to some extent by Newton's law of gravity by virtue of having mass. So the first question we had to ask ourselves is, what are the conceptual equivalencies that underlie all complex evolving systems? For us, evolution occurs in any system that has three properties. Number one, a large number of diverse interacting components. Two, a way for those components to sample myriad configurations. And three, selection for function. Let me give you a few examples to illustrate what I'm talking about. So in nucleosynthesis, our interacting components are protons and neutrons. The ways to sample new configurations of them are through stellar fusion, supernovae, stellar mergers, and there's a selection for stability. In mineral evolution, our interacting components are one level higher. They are the elements of the periodic table. And you sample new configurations of these elements through the various modes of mineral paragenesis. As planets evolve, they subject their materials to new pressure, temperature, composition regimes. And again, there's a selection for stability. In biological evolution, our interacting components are many, and at many scales: genes, cells, species. You sample new configurations through mutations, horizontal gene transfer, recombination, etc., and there's a selection for survival and propagation. At this juncture, it was really important for me to try to address this question of what do we actually mean by selection for function? In other words, what are the most fundamental selection forces in nature? What are the functions that the universe, quote unquote, cares about? And so in the paper, we outline what we think are the three fundamental sources of selection, namely static persistence, which is simply the idea that configurations of matter will persist in time unless kinetically favorable avenues exist for their incorporation into more stable configurations. Static persistence allows there to be out of equilibrium structures in the universe whose dissipation can drive the emergence of dynamic systems, such as hurricanes, convection cells, fire, even life. And in these dynamic systems, processes might emerge, which are defined as any process, functions may emerge, which are defined as any process that has causal efficacy over that system's dynamic persistence. And finally, we think that there is a selection pressure for novelty generation. Why? Because the discovery of new functions that promote dynamic persistence raises that system's quote-unquote kinetic barrier against decay to equilibrium. And we see novelty generation all over life, from the exaptation of one function to another, or even the way that bacteria can tune their mutation rates such that they mutate faster in more stressful environments. And so finally, given this functional view of evolving systems, what might be an auspicious ontology for a law of increasing complexity? Here's where we turn to this idea of functional information, which essentially quantifies the fraction of all possible configurations of a system that possess a certain degree of function. So here you have your entire configuration space. Some of those configurations can perform a specific function. The functional information of the system is defined simply as the negative log base 2 of that fraction of configurations that can perform the function. Given this definition of functional information, why would the functional information of any given system change over time? There are two main reasons why it might change over time. You might have an expansion of your configuration space or strengthening of selection pressures. And to illustrate this, let me turn to an example from the abiotic world, mineral evolution. So in mineral evolution, we have configuration space expansion due to the ever-increasing diversity and availability of elemental components that can comprise minerals, as well as the idea that planetary environments will continuously sample new pressure, temperature, and composition spaces as they evolve. We also have strengthening selection pressures in that there's an ever-increasing time between crystallization of any given mineral in the present, and the fact that new minerals arise by modification of the prior generation of minerals. And in a new paper that we have recently submitted and is under review currently, we've actually quantified the functional information in mineral evolution, showing that there is a monotonic increase in the functional information of mineral systems from the first minerals that were born in stellar atmospheres early on in the universe, all the way through the minerals that are associated with igneous and metamorphic processes associated with plate tectonics on Earth. And so we're proposing, emphasis on the word proposing, a new natural law, the law of increasing functional information, which simply states that the functional information of a system will increase, that is, the system will evolve, if many different configurations of the system are subjected to selection for one or more functions. What are the implications of a natural law such as this? Well, there are many. I'm just going to highlight two to wrap up this presentation. Number one is that we can see Darwinian evolution as a special case of a much more general phenomenon of evolution writ large that can be applied to all kinds of physical and chemical systems, including mineral evolution. Also, to tie us back to the first half of this talk, there may be a distinction between life and non-life that is related to selection for function. And maybe the reason why the machine learning algorithm that we've developed is so good at detecting the difference between life and non-life is because the organic molecules in life have been sculpted by selection for myriad biochemical functions, whereas the organics that we find in, say, meteorites have only been sculpted by selection for static persistence. And so to wrap up the take-home messages today, I hope you've been excited by this new biosignature technique based on pyrolysis GCMS combined with machine learning that achieves 90% accuracy in distinguishing biotic from abiotic samples. And I hope your minds have been tickled, if not expanded, by this proposition for a new natural law based around increasing functional information in complex evolving systems. Thank you very much, and I'd be happy to take your questions.
We have 3 or 4 minutes for questions.
Hi, this is a super cool talk. I'm so excited. I want to talk to you after as well. Hypatia Medavia from Arizona State. Okay, so you've trained this learning algorithm to be able to identify these different samples in biotic and abiotic on Earth. Do you think it would have the same efficacy for samples from a different system that are maybe responding to, maybe they have different functions, maybe they are responding to different selection pressures that have caused them to have different patterns? Do you have any thoughts about that?
Yeah. So let me just back up to this slide. Right here. Yes. So the idea is that, our approach is hopefully agnostic to the specific functions, the specific substrate of the biochemistry that has been sculpted by a completely different trajectory of evolution on a very different world, right? And so the idea is that selection for function will basically move abiotic matter somewhere else in this space. On Earth, it moved it towards here, right, in the, buttons. And then through degradation might, change it by moving it this way in this space. But on another world, it should, it might sculpt it in a very different trajectory. And so the biosignature technique looking at, potentially spotting signs of alien life with very different biochemistries from us is basically an anomaly detection strategy for looking for something with a distribution of organic matter that is very different from all known abiotic samples and has been sculpted by evolution, but maybe in a different trajectory from the exact way that life on Earth has, because that's just one possibility. As somebody was talking about before today, frozen accidents in evolution, right? And so we want to expose ourselves to trying to be able to capture all of the different kinds of frozen accidents that may have occurred on different worlds. And I think by using this method, we'll be able to potentially catch a lot of those different end results of evolution that wound a different way on the respective planets. So thank you for the great question.
I think we are out of time and have to move on to the next person, but I will say there's a break after this where you can go and pick Michael's brain. I hope he's okay with that.
Yeah.
Hello. Can you all hear me? Okay. All right, I think I'm gonna stand over here. So my name is Danny Buckheister. I just finished my second year of my PhD at Penn State, working with Jen McElhatty in the Geosciences department. And I'm gonna be talking to you today, as you might be able to tell from the title, about microbes, caves. But first I wanna acknowledge the fact that I currently am the embodiment of the 20 minutes that separates you from the freedom from those very strange chairs. So in honor of that, I am going to ask everybody, oh, did it give up? You too, probably. I can stand behind the podium, I don't mind.
Okay. I'm sorry.
Oh, it's okay. End of the day, jitters. All right, I'm gonna ask everyone to take a deep breath with me. Let's. Yeah, so I feel pretty powerful right now. No, actually what I wanted was for us all to think a little bit about oxygen. So often we think of oxygen as being pretty synonymous with life. We might think about the oxygen that we breathe, that all the animals around us breathe. Maybe we think of oxygen produced by plants, but what about life before oxygen? And so some of you may be familiar with this sort of figure, but some of you may not be. That solid line there, we're looking at the rise in oxygen in our atmosphere over time, over Earth history. And so if we look down to the Hadean and the Archean when we think that life was emerging, we see that there was very little oxygen in our atmosphere. And so instead of things like oxygen, nitrate, sulfate, things that we find in surface environments today, it was dominated by things like hydrogen sulfide, CO2, methane, iron 2, and things like that. So in this vastly different geochemical environment, a lot of astrobiologists have asked, okay, well, what then were Earth's earliest metabolisms? With this more limited menu, what could microbes at the time do? And so there are a lot of ways to try to go about answering that question, but one of the ways we can do it is through finding analog environments here on Earth, places that look similar to what we think early Earth environments may have looked like. And these environments can be windows into the past. Through finding these environments here on Earth today, we can look at life in them and learn a little bit about the strategies they employ to survive there, and maybe we can sort of trace those things back in time to see if they would have been feasible for early Earth life. And so one of these systems is the Frasassi cave system in central Italy. And so this is kind of an interesting cave system because you have this anoxic sulfidic groundwater from below mixing with this oxygen-rich water coming from above, but also atmosphere. And so you have this geochemical mixing of two very different sort of surfaces clashing, and that creates a lot of interesting geochemical niches that microbes live in. But for the purpose of this talk, I'm going to be focusing on that anoxic, sulfidic groundwater that underlies the system. And that water floods some of these cave passages today. And so about 15 years ago now, deep cave explorers and cave divers discovered a really interesting biofilm in these submerged cave passages. And so imagine you're a cave diver swimming along and you see that. I can't believe they didn't turn around, quite frankly, because it's spooky looking. It sort of hangs down in these ropey, feathery things from any overlying rock. And these rope-like biofilms are full of pyrite. But also, the surrounding water lacks, again, a lot of those common oxidants we think of in surface environments today. So oxygen, nitrate, iron 3 are pretty much all always below detection limits in the water. So in this way, this looks a lot like what we think early Earth environments might have looked like. And this biofilm gives us a really interesting opportunity to look at life and what it can do in this environment, isolated from other potential sources of energy like serpentinization, like photosynthesis. This biofilm is kind of on its own down there. And so in order to study it more fully, we went on an expedition to Italy in February of 2023. And one of the primary goals here was to collect biofilm material for lab-based enrichment cultures. And so in an enrichment culture, you're essentially designing a medium that is supposed to target a metabolism of interest in a lab setting. And so for us, we were looking at a couple different target metabolisms that might be relevant for early Earth environments. So first, we were looking at methanogenesis. Now, there are a few ways of doing methanogenesis, but in this case, we were looking at the combination of hydrogen and CO2 into methane and water. And so this is thought to be a very early metabolism here on Earth. In fact, a lot of people think it's perhaps one of the earliest. And it's relatively well studied in the lab, as opposed to maybe other more rare metabolisms. And so in this case, the goal was just to investigate strategies in an early Earth analog environment. So are these organisms linking up their metabolism with other organisms? Are they forming centrifuges? Do they depend on each other? And do they have any interesting strategies for existing in this relatively low nutrient environment? Now, the second metabolism I was targeting is a little bit different. I'm going to call it here the Vectorshäuser reaction, big, long German name, just like mine. But here we have mineral FeS combining with hydrogen sulfide to form pyrite. That should ring a bell, because I mentioned that that biofilm is full of pyrite, as well as hydrogen gas. Now, this is currently a theoretical metabolism. It was first proposed by a man named Gunther Vectorshäuser in the late 80s, early 90s, as potentially an option for the first metabolism on Earth based on this sort of iron-sulfur world idea that was popular at the time. And it should yield some free energy given thermodynamic calculations, both sort of in ideal conditions, but also in this particular environment. However, it hasn't been seen in any microbes that we know of. So the goal here is a bit simpler. The goal was to identify organisms that potentially gain energy from this reaction. And so to do this, I designed some media, so a very, very limited liquid medium for these microbes to inhabit, and then added the reactants of these two metabolisms into those to try to give them a nice comfy home to want to grow in. So for methanogenesis, that's hydrogen and CO2. And for this Vectorshäuser reaction, that's hydrogen sulfide in the headspace, but also mineral FeS in the liquid itself. And so I want to focus on the methanogens first. So if we think about what we would expect to see in this bottle over time as this reaction proceeds, we would see something like this, where over time you see methane increasing in that gas headspace, and you see hydrogen and CO2 decreasing. And so I was able to monitor these cultures that I started last year through gas chromatography, just the simple one, just GC without the letters at the end. But that allowed us to track this over time. And so taking a look here, we're looking at methane curves over time. On the x-axis, we have the date. And then on the y-axis, we have the peak area for methane. You can think of this as how much methane there is. So the higher the peak area, the more methane there is in the bottle. And then these solid lines are all microbial cultures, so bottles that had microbes injected into them. And then right down at that 0 line, you might be able to barely see it, is a dotted blue line. That represents the abiotic control, so media that did not have any microbes added to it. And so as you can see here, that abiotic control really doesn't go anywhere. It stays at pretty much no methane the entire time. Whereas all of the cultures that have had cells added are increasing in methane over time until they perhaps peak or even out. So that's a really good sign for me, right? Okay, I'm getting methane from organisms that are supposed to make methane, but what about those reactants? And so here we're looking at a very similar sort of plot for CO2 on the left and hydrogen on the right. And so we can see here, although the trend may not be as obvious, by the end of that incubation period, we have a definite fall off in both CO2 and hydrogen in the bottles with life versus the abiotic control, which pretty much stays the same through the experiment. And so these are all really good signs. Methane is increasing, CO2 is decreasing, hydrogen is decreasing. But I wanted to see, okay, do we actually have cells? Are we sure that there are cells in there? And in fact, when we looked, there were cells, and there were quite a few of them. But when we stained them with DAPI, we can see that even better. So that's going to stain cells and hopefully nothing that isn't a cell. And we see a couple different morphologies here, which is really exciting. To verify that these are, in fact, what we think they are, we did some sequencing. And this is very fresh data. For those of you who are into microbes, you can geek out for a second. For those of you who are not, I'll put it plainly. We see that those big blue bars that is dominating most of the cultures here are Methanosarcina, an archaea that was mentioned earlier, and that does in fact make methane through this pathway, taking hydrogen and CO2 and making methane. But we also have a subset of bottles that has another methanogen growing there, a little less common, a little less studied, this Methanocellus. And that also is known to take hydrogen and CO2 and make methane. So far we're really excited about having something growing in the lab. Yay, they're growing. They can survive transfers from one bottle where they have sort of expended their food to another bottle, and they've done that over 3, 4, 5 transfers, which is a really good sign we can keep them growing in the lab. Now the next steps are to try to isolate those strains, characterize them, and potentially investigate their role within the larger biofilm community. Now turning back to the Vectors-Heuser reaction, this is a little bit more difficult. It can be challenging to measure hydrogen sulfide. You can measure hydrogen, as we saw in the methanogens, but something else could be eating that. For example, methanogens would be very happy to gobble that up, so that's not necessarily a great way of tracking a product. And to make all this worse, there are other methods of monitoring, but they're more destructive. The liquid medium is also black, and I mean like jet black, so you can't even look for changes in turbidity, which might suggest to you that these cells are growing. So after about a year of nothing really changing, I was ready to give up on these cells, and then I thought, you know what, let's look under the microscope. And to my surprise, I'm seeing cells. This is that same stain that should stain cells and nothing else. But what was maybe more exciting and made me sort of gasp is this image. And if you are a geoscientist, you might be like, I know what that is. But for many of you, you're like, okay, it's a bunch of black blobs. This shape is fairly characteristic of pyrite framboids. So not only do I think I have cells in this culture, but I also have pyrite, and a good amount of pyrite too. And the cells seem to be pretty closely associated with that pyrite. But before I got too excited, I said, okay, was this maybe just something that came over with the initial inoculation of biofilm material? So we're looking at that culture here, and again, we see those pyrite framboids throughout the frame. And when we look at the abiotic control, we see something very different. Maybe you can pick out a couple blobs that look like they could be pyrite, but all in all, we're seeing pyrite in the microbial cultures and not in the abiotic control. So that's a really exciting thing. There are cells in the culture, likely some of them are active. Pyrite has formed in these cultures over the last year through some sort of mechanism. And it's likely that there's a microbial role in that formation, given that we don't see the same pyrite in the abiotic control. So the next steps here are to look and see how active that culture might be, how long it's taking them to grow, how fast we might be able to get them to grow again. See if we can actually transfer them and keep them growing. Sometimes this is the biggest challenge with culturing in the lab. You get something to grow and then it abruptly dies on you. And we do have sequencing pending, so we can figure out who is in those bottles. I am desperate to know. Some quick conclusions here. Our lab culturing of this unique biofilm is possible on at least months-long timescales, which is very important when you're trying to finish a PhD. Hunting for theoretical metabolisms pretty much has to involve some creative solutions and creative thinking about how you're going to track these down. Lab cultures can, in fact, form pyrite, and that's really exciting because we don't really know the role of microbes in pyrite formation. But also that further field experiments will help us define the system. And very quickly, I do want to say that I recently returned to Italy just last month. We were trying to fill in some gaps here with actually some hydrology, so looking at things like conductivity and depth of the actual water level to see if perhaps there is influx of fresh oxygenated water over time, which really changes the dynamics of the system. But I'm also trying to use the power of the system itself to see if we can grow some of these microbes in capturable forms in its native environment, which looks a lot like that and not like a bottle in the lab. But what this really means is that it's just DIY chaos. If I had known the screen was going to be so big, I may not have put this picture in. But it's a lot of just figuring things out, putting things in weird other things, and sticking it in the water and hoping something grows there. And with that, I would like to thank especially my Italian collaborators. We couldn't do any of this without the cave divers we work with. They are amazing. Great people, and I literally would not be able to study any of this without them. But I'd also like to thank the expert cavers who keep us safe. If you've ever wondered what it was like to do fieldwork in central Italy, this is a little sneak peek of what that looks like. I'd also like to thank my Penn State and US collaborators and my funding support as well. And with that, I will take any questions.
Hi, I'm Chloe Cates from Georgia Tech. This is really, really cool. I was just wondering if you guys not only did sequencing work of your lab cultures, but also of just the environmental samples themselves to see if there are any difference in the enrichment.
Yeah, so we initially sent off some samples from last year for metagenomic sequencing specifically. I was actually planning on talking about that at this conference. Unfortunately, we just got it back and they were like, well, it's 99.1% unclassified. And that seems to be typical for this biofilm, actually. It seems like there is a lot of microbial dark matter and potential for things that we just don't really know too much about. That was, of course, a metagenome, which is a little bit different than the type of sequencing I did on the lab cultures. So we have just sent off fresh sample for 16S, and hopefully that'll provide some more solid identifications of some of these microbes. But all signs from past work and my own work indicate that this is a pretty diverse biofilm with a lot of unknown microbes in there. Which makes it really exciting to investigate further, but also can be very frustrating when you get back like a whole bunch of, we don't know. So yes, pending.
Very cool, thank you.
Thank you.
Final call. All right, thank you for your talk.
Thank you.
Just a little bit of housekeeping before we go on the break. We are on break until 4, so you have 40 minutes. And then 4 to 5:30 is our first poster session. And then at 5:30, we're going to have dinner right out in the poster hall where we also had lunch and breakfast. And there's also a message in the Slack about a possible happy hour tonight. So if you would like to go, please respond with an emoji. Thank you.